Somewhere inside your body right now, your blood is doing what it has always done. Carrying oxygen, fighting off invaders, keeping you alive. You probably never think about it. Most people don't. It's just blood. Red, warm, invisible under the skin. But buried inside that blood is a single biological marker, a tiny protein flag on the surface of your red blood cells, and whether you have that flag or you don't has somehow managed to generate more legends, more conspiracy theories, more wild speculation about alien bloodlines and hidden powers and secret elites than almost any other piece of human biology ever discovered. The question is why? Why does one blood trait spark so many stories? That's what we're here to find out. And here's the thing that I think makes this topic genuinely fascinating, not just as conspiracy bait, but as a real scientific puzzle. There are parts of this story that are completely solid, verified, replicated, peer-reviewed, settled science. And then there are parts that are genuinely uncertain. Not because scientists are hiding something, but because the human past is messy and incomplete and hard to read. And when you sit down and look carefully at where the hard evidence ends and where the speculation begins, you start to understand exactly why a story like this one takes on a life of its own. Uncertainty is the engine that drives mythology. Always has been. Always will be. So let's talk about what we're actually dealing with here. You've heard of blood types before. AB, ABO. That system is called the ABO system, and it describes a different set of markers on your red blood cells. It's the one doctors check before a blood transfusion to make sure they don't accidentally give you blood that your immune system will attack. Most people know their ABO type. Some people have it tattooed. It became cultural shorthand in certain countries for personality and compatibility. Type A supposedly organized and anxious. Type O the universal donor and the natural leader. You've seen the memes. But the ABO system is only one layer of a much more complex picture. The RH system is a separate layer entirely. It's a completely different set of proteins, discovered later, understood differently. And the one that gives us the two letters you've probably seen scrawled next to your blood type on a medical form. That little plus or minus sign. Positive or negative. If you're a positive, your red blood cells carry both the ABO markers that make you type A and the R protein that makes you positive. If you're A negative, same ABO markers, but the RH protein is absent. That absence is what the minus sign means. The protein that most humans have, you don't. And that fact, that single biological absence, is what the next however many minutes of this conversation is entirely about. Now, before you start wondering whether that makes you special or ancient or descended from something not quite human, I want to be upfront with you about how this is going to go. We're going to cover the actual discovery of the RH factor, which has a genuinely interesting story behind it, and a name that turns out to be slightly misleading. We're going to go deep into the genetics, because understanding how RH negativity is inherited tells you a lot about why it exists at all and why it persists in the population. We're going to look at where RH negative blood is more common on this planet, because it is not evenly distributed, and that uneven distribution is one of the things that feeds the legends. We're going to talk about the Basques, a population in southwestern Europe that has the highest rate of RH negativity anywhere in the world, and the theories, both scientific and wildly unscientific, that have grown up around them. We're going to look at royal bloodlines and the idea that certain ruling families track their ancestry with unusual obsession, and what blood might have actually meant to them. And then we're going to walk directly into the myth-making machine, the claims of psychic abilities and heightened intuition and extraterrestrial origin, the YouTube rabbit holes and the forum threads and the New Age traditions that have built entire cosmologies around a blood protein. We're going to take all of that seriously enough to examine it, and then we're going to ask the hard question of what holds up and what doesn't. Here's what I can promise you. I'm not going to tell you that RH negative people are alien hybrids. The evidence does not support that. But I'm also not going to dismiss the genuine scientific mysteries that do exist around Rh negativity, because there are some. The question of why the Rh negative allele exists in such high frequencies in certain populations is a real question. The genetic puzzle of how a seemingly disadvantageous trait survives across thousands of generations is a real puzzle. The story of an ancient population that may have sheltered in isolation during the last ice age and emerged carrying unusual genetic signatures is a real story, even if it's a more complicated and honestly more interesting one than the alien version. Because that's the thing about reality. It tends to be stranger and richer than the myths we build to replace it when we run out of patience for the slow work of actual answers. A population bottleneck, an ice age refuge, a quirk of genetic drift, a cascade of folk legends and royal obsessions and internet acceleration, all of it tangled together across thousands of years into something that feels like a secret. That's more interesting to me than bloodlines from Sirius or underground lizard people, but we'll get to all of it. What I want you to hold on to right now, right at the start, is this. Something genuinely unusual is sitting inside that minus sign. Not supernatural, not proof of ancient alien contact, but real and unexplained enough to deserve a serious look. The kind of look that starts with asking simple questions and follows the evidence wherever it actually goes, even when it doesn't go where the legend wants it to. So that's exactly what we're going to do. We'll start at the beginning with the discovery itself, a lab experiment in the 1930s, a rhesus monkey, and a moment that changed medicine forever. And from there, we'll build outward, from the molecular level all the way to the myth. Before we go any further, though, I genuinely want to know where you are right now. Are you listening to this at 2 a.m. with the lights off, driving somewhere, on a lunch break in a city I've never been to? Drop a comment. Tell me where you are and what time it is there. I look at those comments more than you probably think, and every time I do, I'm struck by the same thing. Wherever you are, whatever time it is, you're carrying your own blood type around with you, and you've probably never once thought about it. Let's change that. So let's start at the molecular level, because that's where the real story lives, and it's actually not as complicated as you might think once you strip away the jargon. Your red blood cells are not smooth, featureless spheres. They're covered in proteins, hundreds of different proteins sticking up from the cell surface like flags on a castle wall. Some of these proteins are just structural, holding the cell together, helping it squeeze through capillaries without tearing apart. But some of them are antigens, and antigens are the ones that matter for this conversation, because antigens are the proteins your immune system uses to identify things, to distinguish self from not self, friend from threat, from when your immune system encounters a foreign antigen, one it doesn't recognize from its own cells, it treats it as an invasion. It builds antibodies against it. It remembers it. And if that same antigen shows up again, the immune response is faster, harder, and potentially dangerous. This is the same basic mechanism that makes vaccines work and that makes certain blood transfusions deadly. Your immune system is essentially running a constant identity check on everything in your bloodstream, and anything that doesn't match the approved list gets flagged for destruction. The Rh system is a collection of antigens. Technically, the Rh blood group system includes over 50 recognized antigens, each one a slightly different protein configuration on the surface of red blood cells. But when most people talk about being Rh positive or Rh negative, they're talking about exactly one of those 50, just one. The antigen called Rhd, or simply the D antigen. That is the entire story behind the plus and minus signs you see on your blood type. Do you have the D antigen displayed on your red blood cells? Then you're Rh positive. Do you not have it? Then you're Rh negative. That's it. That's the whole biological definition. The D antigen is a protein produced by a gene called Rhd. Rh positive people have a functional version of that gene. It produces the protein sits on the cell surface. Done. Rh Negative people either have a deleted version of the gene, meaning the gene isn't there at all, or in some cases a version that doesn't produce a working protein. The cell surface is missing that particular flag, not broken, not damaged, just absent. The machinery for producing one specific protein out of the thousands your body makes every single day simply doesn't run. Now here's where it gets slightly more nuanced, and this is the part that often gets glossed over in casual conversations about blood type. There's a difference between what blood type testing actually tells you and what's happening at the genetic level underneath. When you get tested for your Rh type, the lab is running a phenotype test. It's looking at your red blood cells and asking a simple question. Is the D antigen present or absent? Positive or negative? That's your phenotypic result, and it's the number that goes on your medical file. But your genotype, the actual genetic blueprint, can be more complicated than that, because you inherit 2 copies of most genes, one from each parent. And with the RHD gene, the combinations matter. If you inherited 2 functional copies of RHD, one from your mother and one from your father, you're Rh positive, and you're what geneticists call homozygous for the positive type. If you inherited 1 functional copy and 1 deleted or non-functional copy, you're still Rh positive phenotypically, because the 1 working copy is enough to produce the D antigen and trigger a positive test result. But you're carrying a hidden copy of the negative variant. You're heterozygous. And crucially, you can pass that negative copy to your children. They might test negative even though you tested positive. This is why 2 Rh positive parents can sometimes produce an Rh negative child, and why that result can cause so much confusion and occasionally some very awkward family conversations. If you inherited 2 non-functional or deleted copies of RHD, then you're Rh negative, both phenotypically and genotypically. No D antigen on your cells, no functional gene to produce it. The reason this distinction matters is that simplified testing gives you a simplified picture, and simplified pictures can make things seem more mysterious than they are. The positive or negative label on your blood type is a phenotypic shorthand. It tells you what's detectable on the cell surface. It doesn't automatically tell you what combinations of gene variants you're carrying, or what you might pass to your children, or how your individual immune system will respond in specific medical situations. The biology is more layered than the label suggests. Now, why does any of this actually matter outside of a biology classroom? For the vast majority of your life, your Rh type is medically irrelevant. If you're Rh negative and you never receive Rh positive blood, and you never carry a pregnancy where the fetus has Rh positive blood, then your Rh status will likely never cause you a single problem. The D antigen is just absent from your cells. Your immune system has no particular relationship with it, positive or negative. You don't know it's missing. It doesn't affect how your blood carries oxygen or fights infection, or does any of the things blood is supposed to do. You are, by every measure that matters for daily life, completely fine. The medical significance of Rh status kicks in through a mechanism called sensitization. This is what happens when an Rh negative person's immune system encounters Rh positive red blood cells for the first time. Because the D antigen is foreign, something the immune system has never seen before and doesn't recognize as self, it responds the way it responds to anything foreign. It builds antibodies against the D antigen. It gets sensitized. And from that point on, if Rh positive red blood cells enter that person's bloodstream again, the antibodies are ready. They attack. They trigger the destruction of the foreign cells. For most adults, the most common way sensitization happens is through an incompatible blood transfusion. You give Rh negative blood to an Rh negative patient, no problem. You accidentally give Rh positive blood to an Rh negative patient who has already been sensitized. That becomes a medical emergency. The immune system recognizes the antigen and launches an attack on the transfused cells. This is why blood typing is not optional in emergency medicine. It is not a formality. It is the difference between giving someone life and accidentally triggering an immune assault on what you just gave them. But the context where Rh status matters most, and where it has historically caused the most devastation, is pregnancy. Specifically, pregnancy where an Rh negative mother is carrying an Rh positive fetus. That's the situation where sensitization can happen across generations, where the immune system of one person can affect the survival of another person who hasn't even been born yet. And that's the mechanism that turned Rh blood typing from an interesting scientific footnote into one of the most consequential medical discoveries of the 20th century. We'll get into the full weight of what that means in the next section, because it requires understanding not just the biology, but the history. The story of how anyone figured any of this out at all is a genuinely strange one. It involves researchers working in the late 1930s, a series of experiments on rhesus monkeys, a scientific name that stuck even after the original experiment turned out to be more complicated than it first appeared, and a chain of discoveries that moved fast enough to save lives within just a few years of the first findings. The timeline is remarkable. The science was happening in real time, with real stakes, and the people doing it were working largely in the dark. What they were building, without fully realizing it, was the foundation for every question we're going to spend the rest of this conversation asking. Because before you can ask why Rh negative blood exists, or where it came from, or why it's concentrated in certain populations and absent from others, you first have to understand what it actually is. And now you do. A missing flag on a cell surface. A protein your immune system never learned to recognize because it was never there to recognize. One gene, one antigen, one minus sign, and somehow, through a long chain of history and science and speculation and myth, an entire mystery. The name itself, it turns out, is the first clue that something was more complicated than it seemed. To get why the name is weird, you have to drop into the world of early 20th century medicine, when blood transfusion was still half miracle, half Russian roulette. Doctors already knew about the ABO system. That discovery at the turn of the 1900s had taken transfusion from "let's hope this doesn't kill you" to "we have a fighting chance of not killing you." If you matched A, B, AB, and O correctly, patients stopped dropping like flies on the operating table. It was a revolution. But there was a problem. Even when doctors got ABO right, sometimes things still went catastrophically wrong. Patients who should have been compatible on paper reacted badly to transfusions. Sometimes not the first time, but later. They'd spike fevers, their urine would turn dark, their kidneys would fail. It was clear some invisible compatibility rule was being broken, but nobody knew what it was. So the early transfusion era ran on a mix of boldness and trial and error. You cross-matched blood samples directly, mix a bit of donor blood with a bit of recipient blood in a test tube and watch. Does it clump? That's a no. Does it stay smooth? Maybe it's safe. Not exactly a comforting standard when you're the one on the table, but it was better than nothing. Hidden inside those experiments was the first hint that ABO was not the whole story. Something else, some other antigen system, was quietly operating in the background, invisible to everyone who wasn't looking very, very carefully. The search for that something else really crystallized in the late 1930s, when a pair of Austrian-American researchers, Karl Landsteiner and Alexander Weiner, went hunting for new blood factors using the lab tools they had: rabbits, guinea pigs, and monkeys. Landsteiner already had a Nobel Prize for identifying the ABO groups. He knew from experience that you can discover blood antigens by forcing an immune system to complain about them. Take red blood cells from one species, inject them into another species, and the recipient's immune system will produce antibodies against whatever looks foreign on those cells. Then you test those antibodies on human blood and see what happens. So Landsteiner and Wiener took red blood cells from rhesus macaques, those small monkeys used all over lab research, and injected them into rabbits and guinea pigs. The rabbits dutifully produced antibodies against the monkey red cells. Then came the key step. They took that rabbit serum and mixed it with human blood. Something interesting happened. In about 85% of samples, the human red blood cells clumped together. In the remaining 15%, they didn't. That meant the rabbit antibodies were recognizing a specific antigen that was present on most human red cells and absent from a minority. To Landsteiner and Wiener, this looked like a new blood factor, one you either had or you didn't, analogous to A and B, but clearly distinct. They named the antigen they thought they'd found after the animal that had provided the original red cells, the rhesus monkey, shortened in the literature to RH. In their minds, this RH factor on human cells was immunologically related to an antigen on rhesus macaque cells. It made sense at the time. Name the thing after the experimental system that revealed it. Nobody suspected that this convenient label would later cause decades of confusion, conspiracy speculation, and one of the most persistent misunderstandings in blood group history. Because here's the twist. The D antigen we now mean when we say Rh positive is not actually the same antigen Landsteiner and Wiener thought they were seeing. Later, more refined experiments showed that the factor they detected in the rhesus monkey wasn't identical to the D antigen on human cells. There are Rh-like proteins in primates, but the monkey version and the human D antigen are not simple one-to-one twins. The resemblance is loose, not exact. The original rhesus factor and the modern RhD turned out to be cousins at best, not clones. By the time this became clear, the name had already escaped the laboratory. Clinicians were using Rh factor in blood bank protocols, medical students were memorizing Rh positive and Rh negative for exams, and journals were filled with papers using the same terminology. The term stuck for the simplest possible reason. Changing it would have been a logistical nightmare, and everyone already knew what it meant in practice. Historians of science like to point this out because it's a perfect example of how scientific language is shaped by messy, contingent events, not by some perfect naming committee. A slightly misleading label, born from a specific set of experiments in the 1930s, got locked in because it was clinically useful and easy to say. Rh was short, it fit on blood bank forms, it worked in emergency rooms. Nobody was going to swap it out for something more accurate but more clumsy like de-antigen status once it had woven itself into the daily routines of hospitals. And outside the lab the name took on a life of its own. Tell the public that there's a mysterious rhesus factor in human blood, and some people will jump straight to, does that mean I have monkey blood? If I'm negative, does that mean I'm missing some ancestral primate component everybody else has? This is where language and myth start holding hands. A technical label, originally chosen as a kind of lab shorthand, became a hook for all kinds of imaginative narratives about origin, purity, and otherness. Inside medicine, though, Rh very quickly stopped being an abstract curiosity and became a matter of life and death, because those same years, late 1930s into the 1940s, were when doctors finally connected this new system to something they had been watching helplessly for decades. A devastating condition in newborns. Obstetricians had long seen a mysterious pattern. Some women seemed cursed when it came to pregnancy. Their first baby might be born healthy or only slightly ill, but later pregnancies ended in tragedy. Miscarriages, stillbirths, or infants who died soon after birth. Their skin yellowed, their bodies swollen, their blood badly damaged. The condition picked up a series of names: erythroblastosis fetalis, hemolytic disease of the newborn. Nobody knew why it seemed to worsen with each pregnancy. or why some families were hit again and again while others never saw it at all. As blood group science advanced, researchers started to suspect that incompatibility between mother and fetus might be the missing link. But ABO mismatches didn't neatly explain the patterns. Something else had to be at work, something that could sensitize a mother's immune system in one pregnancy and make subsequent pregnancies much more dangerous. That something else turned out to be the very same RH system Landsteiner and Wiener had opened the door to. Once clinicians had a way to test for RH antigen presence or absence, they realized that most of the worst cases of hemolytic disease clustered in one particular situation: RH negative mothers carrying R positive fetuses. Now the scattered puzzle pieces started to align. The obscure factor named after a lab monkey, the strange incompatibilities in transfusion medicine, the recurring tragedies in maternity wards, they were all facets of the same underlying biology. And that is where the story of RH stops being a niche lab curiosity and becomes a major chapter in 20th century medicine. In the next section, we'll walk through what actually happens in those pregnancies, how an invisible mismatch between one tiny protein in the mother and that same protein in the fetus can turn a hopeful birth into a medical emergency, and how understanding that mechanism changed the fate of millions of children. To really see what's going on, imagine a very specific situation. You have a woman who is RH negative. Her red blood cells do not carry the D antigen. She gets pregnant with a partner who is RH positive, and the fetus, thanks to basic genetics that we'll unpack later, turns out to be RH positive too. From the outside, nothing about this looks strange. The pregnancy progresses like any other. The mother feels movement, goes to her appointments, listens to the heartbeat on the Doppler. But quietly, there's a molecular disagreement forming between her and her baby. It's not about DNA in general. It's not about foreign in some grand cosmic sense. It's about one protein on the surface of red blood cells. Here's the key point. During most of the pregnancy, the mother's and fetus's blood don't actually mix very much. They're connected by the placenta, which acts like a filter and a trading post. Nutrients and gases cross, but whole red blood cells mostly don't. The immune system on both sides is being carefully insulated. The real drama tends to happen at the border crossings, moments when that barrier gets breached. The most common of those is birth. As labour progresses, as the placenta starts to shear away from the wall of the uterus, as tiny blood vessels rupture, a small number of fetal red blood cells can leak across into the mother's circulation. If the baby is Rh positive, those cells carry the D antigen the mother's cells lack. To her immune system, those red blood cells look off, not like hers, not immediately like enemy combatants, but suspicious enough to get noticed. The first time this happens, her immune system usually responds cautiously. It's like a border guard writing up a report rather than calling in an airstrike. Her B cells start the process of recognizing the D antigen, presenting it to helper T cells, and eventually making antibodies against it. These early antibodies are typically IgM type, bulky, less efficient at crossing the placenta. They mark the beginning of a memory response. Clinically, that's why the first pregnancy is often spared the worst of the damage. The baby is delivered, maybe a few cells sneak across, the mother's immune system takes notes and files them away. The child may have mild jaundice, maybe nothing noticeable at all. Life goes on. But the mother has been changed. Over the weeks after exposure, her immune system class switches. It starts producing IgG antibodies against the D antigen. These are smaller, more refined, and crucially, they move across the placenta very easily. That's usually a helpful feature. Maternal IgG gives newborns a protective shield against infections for the first few months of life. But in this specific scenario, it sets up a dangerous trap for the next Rh-positive fetus. Now picture a second pregnancy, again with an Rh-positive fetus. This time, when even a tiny amount of fetal blood touches the mother's immune system, maybe from small leaks in the placenta, maybe from a minor trauma, maybe from a medical procedure, her body doesn't have to start from scratch. The memory B cells are already primed. They know the D antigen, they know what to do. They switch into high gear. She begins producing anti-DIgG antibodies, much faster and in much higher amounts. Those antibodies circulate in her blood, cross the placenta, and enter the fetal circulation. Once inside, they do what antibodies are built to do: bind to their target. In this case, they stick to the D antigen on fetal red blood cells. Bound up like that, the fetus's red blood cells become targets for destruction, mainly in the fetal liver and spleen. The cells get broken down faster than the fetus can replace them. This is hemolysis, the shredding of red blood cells. From the fetus's perspective, this is a slow-motion disaster. Red blood cells carry oxygen. Destroy enough of them, and you have anemia. Severe anemia pushes the fetal heart into overdrive, trying desperately to move what oxygen is left. Tissues start to swell. Fluid leaks into body cavities. Organs strain to keep up. At the same time, all those destroyed red blood cells leave behind heme, which gets turned into bilirubin. Before birth, the placenta helps clear some of that. After birth, the newborn liver suddenly has to handle it alone. If bilirubin levels soar, it can deposit in the brain, causing permanent damage, a condition called kernicterus. This whole spectrum, anemia, swelling, jaundice, organ strain, is what doctors mean by hemolytic disease of the fetus and newborn, or HDFN. In older literature, you'll see erythroblastosis fetalis, referring to the immature red blood cell precursors churned out in a failing attempt to keep up with the destruction. Before the Rh story was understood, this was a medical horror show. Women would have a first healthy baby, then a second pregnancy that ended in stillbirth, then a third where the infant was born profoundly swollen, jaundiced, gasping for life. Obstetricians saw the pattern, but had no effective way to break it. Whole families were marked by it. Once Rh incompatibility was identified as a major cause, though, a new question emerged. Could you stop the mother from becoming sensitized in the first place? The answer turned out to be one of the most elegant tweaks to the immune system in clinical history. Rh immunoglobulin, often called RHIG, or by brand names like Rho-D immune globulin. RHIG is essentially a concentrated solution of anti-D antibodies, harvested from donors and purified. On the face of it, that sounds backwards. If the mother's own anti-D antibodies are the problem, why would you give her more? Because timing is everything. If you give an Rh-negative mother RHIG at specific moments, most crucially around 28 weeks of pregnancy, and within about 72 hours after delivering an Rh-positive baby, those donor antibodies will bind up any fetal Rh-positive red cells that have slipped into her circulation before her immune system gets a good look at them. They act like a cleanup crew. They tag and clear those cells so fast that the mother's B cells never fully activate, never build that long-term memory, never ramp up their own anti-DIgG production. From the immune system's perspective, the incident barely registers. No file gets opened, no wanted poster goes up. Prevent sensitization once, and you don't just protect that baby, you protect future pregnancies. In places where Rh screening and RHIG prophylaxis became standard, starting in the late 1960s and 1970s, the impact was dramatic. Rates of severe Rh-related hemolytic disease plummeted. What had been a common lethal condition turned into a largely preventable complication, at least in settings with access to prenatal care. That's not to say every detail is nailed down or that every case is simple. Medicine never is. It's widely accepted routine clinical practice that Rh-negative pregnant women are tested and monitored. Rhig is given at specific gestational ages, after miscarriages, abortions, certain prenatal procedures, or any trauma that might cause fetal maternal bleeding, and after delivery if the baby is Rh positive. This strategy drastically reduces maternal sensitization and subsequent HDFN. Those are not controversial statements. They're standard obstetric guidelines in most of the world. Where the research frontier still exists is in the fine print. How much fetal blood exposure is enough to sensitize a particular woman? Why do some Rh-negative mothers get sensitized despite prophylaxis? How do other blood group antigens, Kell, Duffy, Kidd, contribute to similar diseases even when Rh is matched? Can we refine treatments for fetuses already affected with better in utero transfusions or immune modulation? There's also interest in the broader immunology. Pregnancy itself is a kind of controlled immunological paradox. The mother tolerates cells that are half not hers without going on the attack most of the time. Understanding why that truce usually holds, and why it sometimes fails in specific antigen systems like Rh, is still an active area of basic science. But on the big picture, the consensus is clear. Rh incompatibility is not a curse, not a marker of alien blood, not a sign of some metaphysical incompatibility between lineages. It's a very specific immune mismatch around a single protein, operating according to the same rules that govern organ rejection, allergy, and autoimmunity. And yet, the emotional weight of that mismatch, who loses babies, who doesn't, which families carry those stories, has made Rh status feel far more than technical to many people. It's become a narrative about fate, lineage, even destiny. If you're crafting a mythology around bloodlines, you can see why this would be irresistible material. A trait that can turn reproduction itself into a battlefield sounds like something out of myth. But to understand where those stories go, and how far they drift from the underlying genetics, we need to look at what negative actually means in your DNA, and how it ended up in the human population in the first place. To get there, we have to do something people hate doing in school, basic genetics. But stay with it, because hidden in these simple rules is the reason Rh-negative exists at all, and why it behaves the way it does in families. Start with the core idea. For the RhD protein, you don't have Rh-positive genes and Rh-negative genes floating around independently. You have versions of the same gene called alleles. At the Rhd spot in your DNA, you inherit one allele from your mother and one from your father. You can think of them as 2 pages in a recipe book that sits in each of your cells. If a page contains instructions for making the D protein, that's a D allele. If the page is blank where that recipe should be, because of a deletion or a big mutation that breaks the recipe, that's effectively a D allele. Put those 2 pages together and you get 3 basic combinations. D plus D, D plus D, D plus D, in almost all standard lab tests. If you have at least one working D page, D plus D or D plus D, your red blood cells show the D antigen on their surface. You'll be typed as Rh positive. Only if you have 2 broken pages, D plus D, do your cells completely lack that D antigen. That's when you're typed as Rh negative. This is why an Rh negative person can have an Rh positive child without anything mystical going on. Imagine a father who is D positive D. He's Rh positive because one working copy is enough. The mother is D plus D, fully Rh negative. Each pregnancy, the child gets one allele from each parent. The mother can only give D, because that's all she has. The father flips a kind of genetic coin. Sometimes he passes D, sometimes D. So, If the child gets D from dad and D from mum, they're D plus D, Rh positive. If the child gets D from dad and D from mum, they're D plus D, Rh negative. On average, that's a 50-50 chance with this pairing. Two Rh positive parents can also have an Rh negative child if both are D positive D and both happen to pass along their D. Two Rh negative parents, both D positive D, will only ever have Rh negative kids because there's no D to inherit. Once you see it laid out this way, the pattern looks very normal. Same logic as brown versus blue eyes in the old textbook examples. Same Mendelian coin flips. The RhD story doesn't need aliens to produce the surprisingly Rh negative child in a mostly positive family. It only needs a silent D allele carried unnoticed for a few generations. But that's the clean, simplified version. Reality, as usual, is messier at the molecular level. When people say Rh negative, they usually mean no detectable D antigen with standard blood typing reagents. But that no D result can come from more than one underlying molecular cause. In many Europeans, being Rh negative really does correspond to a large deletion of the RhD gene. The page in the recipe book is literally missing. No instructions, no protein. Elsewhere in the world, though, you see something different. The RhD gene is still there, but it's been altered by point mutations, small insertions, or rearrangements, so that the D protein is produced in a scrambled, partial, or reduced form. Those variants can lead to what's called weak D or partial D. People with some weak D variants have a much smaller number of D antigens on each red blood cell. In a routine blood typing test, that faint signal can be easy to miss, especially with older methods. They might be called Rh negative in one lab and weak D positive in another. Others produce a D-like protein that's missing certain pieces. To a lab reagent, those cells may look positive. To a human immune system, certain missing or altered segments can make them behave differently in transfusions or pregnancy. That's why there are long technical tables in transfusion medicine guidelines about which weak D types should be managed as Rh positive and which as Rh negative, especially for pregnant women. The point isn't to drown in the subtypes. It's to see that the positive versus negative label is a crude summary painted over a surprisingly diverse landscape of underlying genetic changes. Sometimes the gene is gone, sometimes it's there but turned off. Sometimes it's there and working at half capacity. Sometimes it's changed just enough to confuse the tests. From an evolutionary perspective, all of these are just different alleles in the population. Different pages in the recipe book, some more legible than others. Now pull back from the individual and look at the population. Every generation, people have children. Those D and D alleles get shuffled, recombined, and passed on. Whether a particular allele becomes more common, stays at the same level, or fades away depends on something geneticists call allele frequencies. If you went out and sampled 1 million people and counted how many D alleles and how many D alleles were floating around in that gene pool, you'd get a ratio, maybe 85% D to 15% D in one region, maybe 60 to 40 in another. That ratio is the allele frequency. Basic population genetics tells us that if people mate more or less randomly with respect to this gene, there's no major survival advantage or penalty associated with D or D, and the population is large, then those allele frequencies tend to drift slowly. The D allele won't suddenly vanish just because it's recessive. Carriers, D plus D, silently move it from generation to generation. That's why a trait can be rare at the visible level. Only D plus D show the Rh negative phenotype, while the underlying allele is fairly common. Add in real-world complications, migration, population bottlenecks, isolated groups marrying mostly within themselves, and you start to see how patches of high Rh negative frequency can appear. Not because of magic, but because of the arithmetic of who married whom, where, over thousands of years. So where does natural selection come in? Has being Rh negative ever actually helped anyone survive? This is where the speculation ramps up and the evidence gets thin. On the harm side, we do have clear, direct selection pressure against Rh negative mothers carrying repeated Rh positive pregnancies in pre-modern settings. Before RHIG existed, families where an Rh negative woman kept conceiving Rh positive babies would, on average, lose more infants after the first sensitizing pregnancy. Over many generations, that kind of reproductive cost should push the D allele downward, and yet Rh negative hasn't vanished. In some populations it's relatively common. That suggests one of several things. The negative selection pressure was weaker than it looks, because not every Rh negative woman had multiple Rh positive pregnancies. Cultural patterns, who married whom, how many pregnancies survived for other reasons, blurred the effect. Or there might have been counterbalancing advantages in some environments. People have proposed all sorts of candidate advantages. Maybe Rh negative individuals were a bit more resistant to certain pathogens. Maybe some forms of malaria or other blood-related parasites didn't like the missing D antigen. Maybe there were subtle effects on immunity or pregnancy that helped in other circumstances. Right now, these ideas are mostly hypotheses. There is some research hinting at associations between Rh status and risks for certain infections or conditions, but the signal is tangled, small, and far from definitive. It's not like the sickle cell trait, where the connection to malaria resistance is strong and well documented. What we can say safely is this: for Rh negative to reach substantial levels in certain regions, you don't need a huge survival advantage. You just need the D allele to avoid being strongly punished, and you need the usual historical forces, small founding groups, geographic isolation, random drift, to have time to work. Imagine a small group carrying an unusually high number of D alleles taking refuge in a valley during an ice age, or crossing into a new region and staying relatively endogamous for centuries. Their descendants will, by sheer genetic bookkeeping, show more Rh negative individuals, no additional magic required. But geography does matter, and the distribution of Rh negative is anything but uniform. Some parts of the world have almost none. Others, like certain pockets of Western Europe, have strikingly high frequencies. That patchwork is where the story starts to pick up that conspiratorial sheen. Why here and not there? Who are these people, and why do their descendants carry so much negative? To answer that, we need to get out of the bloodstream and start moving across maps, following migrations, refuges, and the echo of ancient population crashes that still shape who carries which alleles today. If you could hover above the planet and see Rh negative as a kind of heat map, the picture wouldn't be a gentle gradient. It would be patchy, uneven, almost suspicious looking. Start simple. At the global level, most humans are Rh positive. On average, Rh negative sits somewhere under 20% of the population in any given region, often far below that. In large parts of East Asia, East Africa, and indigenous populations of the Americas, Rh negative is rare, single digits, sometimes edging towards zero. In some communities you can test hundreds of people before you find even one person who's D plus D. Move into Europe and the colours on that imaginary map start to shift. Across much of Western and Central Europe, Rh negative becomes noticeably more common. In many countries you're looking at ballpark figures in the low teens, creeping toward 15%. Not half the population, not anything close to a majority, but enough that if you stand in a crowded subway car, you can be pretty sure a few fellow passengers lack the D antigen completely. Then there are the hotspots, pockets where the proportion of Rh negative climbs much higher than the surrounding regions, parts of the British Isles, sections of France and the Western Pyrenees, small communities in North Africa with European admixture, tiny islands in the global sea of Rh positive. This is the kind of pattern that makes the conspiracy-minded sit up and start connecting dots. Why there? Did something different happen in those places? Are these isolated remnants of an ancient bloodline clinging to their distinctive marker while everyone else diluted theirs away? To answer that, you have to think the way population geneticists think. Less like a novelist hunting for secret lineages, more like an accountant tracking probabilities through time. Three main forces can create these uneven patches without invoking anything supernatural: founder effects, genetic drift, and bottlenecks. Take founder effects first. Imagine a small group of people breaking off from a larger population and settling somewhere new, a valley, an island, a highland refuge. Just by chance, that small group doesn't carry exactly the same mix of alleles as the big population they left. Maybe, in that founding group, the D allele happens to be a bit more common. Nobody notices at first. There are no Rh negative villages, no shrines to the missing D antigen. It's just one trait among thousands floating along in the gene pool. But that founding group grows. Their descendants largely marry among themselves for generations because of geography, language, culture, or simple isolation. Over time, that slightly elevated D frequency in the founding generation translates into a visibly higher proportion of Rh negative individuals in the descendant population. We see versions of this all over human history. Shipwrecked crews becoming the ancestors of island communities, refugees from wars or plagues huddling in mountain valleys, Ice Age bands pushed into last remaining pockets of habitable land when glaciers marched across Europe and Asia. Every one of those events is an opportunity to reshuffle allele frequencies. If the founders of a refuge carried more D alleles than average, their descendants will too. No special destiny required, just arithmetic. Genetic drift is the second ingredient. Even if nothing dramatic happens, no heroic migration, no catastrophic plague. Small and medium-sized populations don't behave like perfectly mixed soup. They behave like a series of coin flips. Each child is a genetic sampling event. Sometimes, just by chance, more D alleles get passed on in one generation than in the previous one, sometimes fewer. Over long stretches of time, especially in smaller populations, this random walk can push allele frequencies noticeably up or down. If selection isn't strongly punishing or negative, drift is free to wander. In one valley, the D allele slowly drops. In another, it quietly climbs. After a few hundred or a few thousand years, you end up with villages, regions, even whole cultural groups whose Rh negative frequency is significantly higher than their neighbours, solely because of drift piling on top of founder effects. Then there are bottlenecks, the big reset buttons of evolution. A bottleneck is any event that slashes a population down to a tiny fraction of its previous size. Drought, epidemic, volcanic winter, war, or a combination of all of them. We talked earlier about one massive species-wide bottleneck almost 1 million years ago, but smaller, regional bottlenecks have been happening constantly. When a population crashes from, say, 50,000 people to 2,000 survivors, it doesn't carry all of its original genetic variation through the eye of that needle. Whatever mix of alleles the survivors happen to have becomes the new baseline. If, by coincidence, those survivors include a few more D alleles than the pre-crash average, the post-bottleneck population will be permanently tilted toward higher Rh negative once it expands again. Layer these processes over the geography of the last ice age, and a pattern starts to emerge. During the coldest periods, much of northern Europe became uninhabitable. Human groups retreated into refuges, warmer, more stable pockets in places like the Iberian Peninsula, southern France, parts of Italy and the Balkans. Those refugees were classic bottleneck zones. Fewer people, more isolation, intense local drift. When the ice retreated, those refuge populations expanded back out. repopulating northern and western Europe. They didn't all carry identical genetic packages. Some had, by chance and by history, ended up with more D alleles. As they spread and mixed, they seeded higher Rh negative frequencies in the regions they came to dominate. So when you look at modern maps and see Western Europe, and especially Southwest Europe, glowing more brightly in arch-negative terms, you're probably seeing a long, faint echo of those Ice Age refuge dynamics, layered with later migrations, invasions, and intermarriages. Now, here's where things can go off the rails if you're not careful. It is easy, almost irresistible, for humans to look at those high-frequency zones and start telling identity stories. We are the negative blood people. Our ancestors were different. This shows we're not like the others. You can see the appeal. Blood feels intimate. A trait that's rare globally but relatively common in your group is a perfect seed for a myth of specialness. And because Rh-negative can have real medical consequences, it feels weighty in a way that, say, attached earlobes don't. But correlation is not destiny, and it is definitely not a moral category. If a mountain community has more Rh-negative than the global average, that does not mean they're purer, older, chosen, or closer to some vanished superior race. It means that across thousands of years, founders, drift, and bottlenecks stacked the genetic deck in a slightly different way. Other parts of their genome will be different in other, far less glamorous ways. Shorter or taller on average, more or less lactose tolerant, a little better or worse at metabolizing certain plant toxins or fats. Rh-negative is one marker on a massive tangled tapestry of variation. Focusing on it and building an entire identity, or worse, a hierarchy, around it is like picking one thread and declaring it the only one that matters. Still, the hotspots are real. Some of them have been the focus of more speculation than others. One in particular has become almost legendary in Rh-negative lore. A population often described as Europe's oldest, most mysterious, and most pure surviving group. A people whose language doesn't seem to belong to any known family, whose homeland sits right in one of those Ice Age refuge zones, and whose Rh-negative levels are famously, sometimes exaggeratedly high. You already know the name, the Basques. To see how science, history, and myth get braided together around Rh-negative, we have to go there next, into the valleys and coasts of the Western Pyrenees, where a real genetic signal meets a century of storytelling. If you wanted to build a legend around a people, you could hardly ask for better raw material than the Basques. Start with the basics. The Basque country straddles the Western Pyrenees between what is now northern Spain and southwestern France. It's a compact region. Coastal bays, steep green valleys, mountain passes, and for a very long time it has been fiercely itself. The standout feature is language. Basque, or Euskara, is what linguists call a language isolate. It's not Romance like Spanish or French. It's not Celtic, not Germanic. It doesn't fit into the Indo-European family at all. For centuries, scholars tried to tie it to everything from Iberian to Berber to Georgian. So far, nothing has stuck. Whatever roots Euskara has, they seem to predate the wave of Indo-European languages that rolled across most of Europe in the last few thousand years. Historically, the Basques also look stubbornly continuous. When Romans arrived, they found tribal groups in the region that look, from the scraps of record we have, like ancestral Basques. When Visigoths, Franks, Moors, and later Spanish and French kingdoms surged back and forth, the Basque valleys remained comparatively autonomous, negotiating, resisting, cutting deals, but not dissolving as a distinct people. Add in a long maritime tradition, Basque whalers in the North Atlantic, Basque fishermen off Newfoundland before most Europeans knew it existed, and you get a culture that is both locally rooted and quietly everywhere, without losing its sense of difference. Now lay the blood data on top of that. In many Basque populations, Rh-negative frequencies are among the highest recorded anywhere. Numbers vary by study and by village, and they're often exaggerated online, but it's not unusual to see figures in the 25 to 30% range for Rh-negative individuals. That's roughly double what you find in many neighbouring European regions, and far above the global average. Combine an ancient-sounding language, a reputation for being old-stock Europeans, and a real measurable bump in Rh-negative, and you have the perfect fuel for a particular kind of story, the living relic population. This is where older anthropology steps onto the stage. In the late 19th and early 20th centuries, physical anthropologists were obsessed with classifying Europeans into races and types. They measured skulls, noses, height, hair colour. They tried to map language, culture and racial type onto each other, often with a heavy dose of bias and wishful thinking. Basques fascinated them. Here was a people whose language didn't fit the Indo-European pattern, living in one of those supposed Ice Age refuge zones, with certain physical traits some authors claimed were distinctive, and later, with unusual blood group frequencies. It was irresistible. By the mid-20th century, once ABO and RH blood groups were understood, Basque samples started showing up in blood surveys. Researchers noticed the high O type and the high Rh negative compared to many neighbours. Some interpreted this as evidence that Basques were a kind of purer pre-Indo-European population, less admixed, closer to the ancient hunter-gatherers who had sheltered in Iberia during the last glacial maximum. Those early narratives had a logic, but they were built on very limited data. A few blood group systems, some craniometry, and a map of where people happened to live at the time. From that thin base, a huge edifice of speculation grew. Outside academia, the story mutated. If Basques were older or less mixed, then perhaps they were survivors of an ancient master race. Perhaps their unusual Rh-negative levels mark them as descendants of Atlanteans, or Cro-Magnon elites, or even something non-human. Books and fringe documentaries started linking Basques, pyramids, megaliths, UFOs. Whatever you wanted, you could plug it into the Basque mystery. The leap is always the same. From this population looks somewhat distinctive, to this population must be radically qualitatively different in origin. From interesting outlier to living relic. Modern genetics has complicated that picture, in some ways supporting a kernel of it, in other ways blowing it apart. Whole genome studies of European populations do show that Basques are somewhat distinctive. When you plot genetic variation across Europe, Basques tend to cluster together and stand a bit apart from many of their neighbours. They show continuity with ancient DNA from the Western Pyrenees and surrounding regions. There's evidence that they have a strong component of ancestry related to early European farmers and local hunter-gatherers, with somewhat less of the later steppe-derived ancestry that spread with Indo-European languages. In plainer language, Basques look like a population that has been sitting in that corner of Europe for a long time, mixing mostly with each other and with nearby groups, and not absorbing quite as much of certain later migrant waves as other Europeans did. But, and this is crucial, they're not a separate species. They are not the last pure representatives of some lost non-human lineage. Their genomes show the same broad mix of ancient European components you see elsewhere, just in different proportions. They have plenty of shared ancestry with other Iberians, with French populations, with Europeans more broadly. Rh-negative is part of this story, but only part. It sits on top of the same population processes we talked about earlier, founder effects, drift, and regional bottlenecks. If you imagine Ice Age bands clustering in refuges, the Western Pyrenees and surrounding coasts are exactly the kind of place that would have hosted long-term groups. Those groups then feed into post-Ice Age re-expansion. If the allele frequencies in those refugees tilted toward more D alleles for whatever historical reasons, you'd expect their descendants, the people still living there millennia later, to show higher H negative rates. Add relative linguistic and cultural isolation, which can track loosely with genetic isolation, and you get a population where certain variants, including Rh negative, stay elevated. Not because they're sacred relics, because the mixing has been somewhat less intense, or has followed slightly different paths than in regions that saw more massive migrations and conquests. From the outside, though, we speak a unique language, and we have unusually high Rh negative. Doesn't sound like a boring tale of founder effects and gradients. It sounds like a story with an aura, a hint of hidden ancestry, a suggestion that this group remembers in its blood a time before everyone else. That's how a real statistical pattern becomes a mythic origin story. Step 1, notice a difference. Step 2, connect that difference to identity. Step 3, back project it into deep time, often far beyond what the evidence can support. Step 4, layer on whatever myth you already want to believe. Ancient astronauts, drowned continents, secret priest kings. Science offers a more grounded but still fascinating alternative. When geneticists look at Basques today, they see one thread in a much larger tapestry of European history. They see signals of Ice Age refuges, Neolithic farmers moving in from the Near East, steppe herders pushing west, local hunter-gatherers hanging on in pockets and blending in various ratios. They see languages rising and falling, sometimes leaving no modern descendants except an isolate like Euskara. They see exactly the kind of messy overlapping migrations and mixtures you'd expect over tens of thousands of years. What they don't see is a clean, unbroken line back to some wholly separate origin. But even that more modest picture carries a hook for the imagination. If Basques preserve, in their genes and language, an unusually strong echo of ancient Europe, what about everyone else? How many other populations, with their own quirks and hotspots and isolates, are faintly mapping old migrations we've barely begun to reconstruct? To answer that, we have to zoom out again, from one mountain people to the whole continent, from a single blood group to the shifting human rivers of the Ice Age and after. Because the Basque case is not an exception to the rule, it's a window into it. And that takes us into the deeper story of ancient migrations, glacial shelters, and how countless small local histories layered together to create the patchwork of auric frequencies we see today. To really understand why Art Negative looks the way it does in Europe, you have to picture the continent not as a static map of nations, but as a breathing, shrinking, expanding organism under the Ice Age. Roll the clock back to around 25,000 years ago, during the last glacial maximum. Enormous ice sheets cover northern Europe. Glaciers crawl down from Scandinavia. Much of what is now Britain, Germany, Poland. Forget farmland and cities. You're looking at tundra, permafrost, environments where only the toughest humans can hang on at all. Most people don't. Human groups in Europe retreat southward into pockets of relative warmth and stability, what scientists call glacial refugia. The classic ones are Iberia and the Pyrenees, the Italian peninsula, the Balkans, parts of the Black Sea coast. Think of them as lifeboats bobbing along the frozen edge of the ice. In those lifeboats, small bands of hunter-gatherers survive, generation after generation. They hunt reindeer and wild horses on the steppe-like plains. They paint caves, carve figurines, make tools. Crucially, they're genetically isolated from one another for long stretches of time. Now bring in what we already talked about earlier in the script. Small, scattered populations, founder effects drift. If one refuge happens to have slightly more carriers of the RHD negative allele, maybe because a few early individuals there carried it, there's no giant mixing bowl to instantly dilute that signal. Inside that pocket over thousands of years, chance can nudge the frequency up or down, just like it did in those older bottlenecks. Meanwhile, another refuge might drift in a different direction. Then the climate starts to shift. After about 19,000 years ago, the ice begins its slow retreat. Glaciers pull back, plants move north, herds follow the grass. And the people in those refuges start to move with them, fanning back out into territory that had been uninhabitable for millennia. This is the first big post-Ice Age re-expansion. Western hunter-gatherers and related groups in other refuges spreading into France, Germany, Central Europe, the British Isles as the ice margin recedes. Archaeologists can track this in the stone tools, in the art styles. Geneticists can see it in ancient DNA, a broad related cluster often called Western hunter-gatherer ancestry, moving out from southwestern Europe and beyond. If you imagine genes like coloured dyes in water, each refuge is its own little vial. When the glaciers melt and populations expand, those vials spill and mix over the landscape, overlapping, swirling, sometimes staying concentrated in certain regions, sometimes blending until they're barely distinguishable. Rh-negative status isn't a dye on its own, but its underlying D allele is part of that swirling pattern. But the hunter-gatherer story is only the first layer. Around 8,000 to 9,000 years ago, a very different kind of human population starts pushing into Europe from the southeast. Early farmers from Anatolia and the Near East. These groups have domesticated wheat, barley, goats, sheep. They move along the Mediterranean coasts and up the Danube corridor, bringing with them a completely different way of life and a somewhat different genetic profile. They don't just sweep the hunter-gatherers away. In some places there's resistance, in others there's coexistence. Over centuries the two lifeways blend in 100 local ways. Hunter-gatherer bands may adopt bits of farming. Farmer villages may intermarry with local foragers, bringing in their genes and their knowledge of wild resources. Genetically, this creates the second major component of European ancestry. Early European farmer. In most modern Europeans, you can still see this layer. It's like reading a faint inscription under newer writing. Different regions preserve different ratios. Sardinia, for instance, holds a relatively strong early farmer signal. Some northern and eastern areas show more of the older hunter-gatherer ancestry clinging on. Now one more big wave rolls in. About 5,000 years ago, populations associated with the Pontic-Caspian steppe, north of the Black and Caspian Seas, begin moving west and south. They probably spoke early Indo-European languages. They may have had horses, wagons, new forms of social organization. Genetically, they bring in a third major component called steppe, or Yamnaya-related ancestry. This steppe-derived ancestry spreads rapidly across much of Europe, especially in the north and west. It doesn't erase what was there. It layers on top of and mixes with the earlier hunter-gatherer and farmer components. In some places, like parts of Central and Northern Europe, the steppe signal becomes very strong. In others, like certain coastal and mountainous pockets, it's more muted. By the time we reach the Bronze Age, Europe is already a palimpsest. Ice Age refuge descendants, Neolithic farmers, steppe herders, and many smaller local groups, all mixing in different proportions from valley to valley, coast to inland plain. This is what geneticists call admixture. In everyday terms, admixture just means populations that were once somewhat distinct start having children together. Over time, their genetic differences get blended. Instead of lineage A over here and lineage B over there, you have people who are partly A, partly B, with those proportions varying across families and regions. That has a very simple consequence. Single origin stories almost always fail. If you want to say R set negative comes from X ancient group, you immediately run into a problem. Which X? The hunter-gatherers in Iberia, the ones further east, the early farmers from Anatolia, some subset of the steppe herders, A mysterious lost Cro-Magnon elite? The reality is more mundane and more interesting at the same time. RHD alleles have been around in human populations for a long time. The D allele that produces Rh negative phenotype is just one variant among many. As different Ice Age refuges and later Neolithic and steppe groups formed, shrank, expanded and mixed, the frequencies of that variant rose and fell locally. In one scenario, Western hunter-gatherers emerging from the Franco-Cantabrian refuge, roughly the Iberia southern France zone, might have had a slightly higher starting proportion of the D allele than some eastern refuges. As they re-expanded north and east into Europe, they could have seeded that variant across large areas. Then, as early farmers moved in, they might have brought somewhat different frequencies, maybe lower on average. When they had mixed with local hunter-gatherers, some regions would have ended up with intermediate levels, others skewed toward one side or the other. Add the later steppe groups, who again had their own baseline frequencies, and you have a three-way blend shaping the RHD landscape. In another, not mutually exclusive scenario, some of the modern Rh negative hotspots, like parts of the Western Pyrenees, reflect not just what came out of Ice Age refuges, but what happened afterward. A valley where, by chance, a few influential lineages carried the D allele. A history of relative isolation, less inflow from steppe-heavy regions. Several thousand years of people mostly marrying within nearby communities. That's exactly the kind of setting where a variant can stay elevated, even if the surrounding areas shift in other directions. Could natural selection also have nudged things in certain places? Maybe. It's plausible that in some past environments, say with particular infectious diseases or reproductive pressures, being Rh negative or being heterozygous with one Rh positive and one Rh negative allele conferred a small advantage or disadvantage. There are hints and hypotheses, but nothing like a simple universal rule. Whatever selection did, it would have been layered on top of the existing patchwork of drift, founder effects, and migrations. What we don't see is a clean, continent-wide signature that screams, here is the one place, at the one time, where Rh negative began and then spread unchanged. Instead, we see a lattice of contributions. Ice Age hunters, Neolithic farmers, steppe pastoralists, later regional movements, all stirring the pot. And that messy reality is exactly what makes simple narratives so tempting. When you're faced with this kind of complexity, a Europe built from at least 3 major ancient populations plus countless minor ones, it's very human to want an anchor. A story that says, this bloodline is from the first people, or these genes are older and purer, or this group has something special that others lost. Rh negative, because it has a striking distribution and real medical consequences, becomes an easy candidate for that role. Tie it to Ice Age refuges, to cave painters, to original Europeans, and suddenly you're not just talking about an allele frequency. You're talking about destiny, about inheritance, about who was here before everyone else. That's the pivot point. From the science side, we have glaciers, refuges, expansions, admixture, a churning population history where no group is untouched by mixing and movement. From the cultural side, we have a deep desire to turn that churn into lineage charts, chosen peoples, and ancient bloodlines. In the next part of this story, we'll watch what happens when those two worlds collide. When royal genealogies, national myths, and occult traditions start to latch onto real, but subtle, biological patterns like Rh negative, and inflate them into symbols of right to rule, spiritual power, or even non-human origin. If you want to see how that collision plays out, you don't start in a lab. You start in a throne room. For most of recorded history, people didn't have DNA tests or blood typing, but they absolutely had an obsession with blood. When a medieval chronicler says a king has noble blood, they don't mean hemoglobin levels, They mean legitimacy, a right to rule baked into the body, passed down through flesh and bone. Blood becomes a shorthand for everything you can't see directly: ancestry, honour, divine favour, even moral character. You can watch this metaphor harden into political reality. In monarchies from Europe to Asia to Africa, the idea of royal blood is treated like a physical substance that can be diluted, contaminated, or purified. Marry the wrong person, and you taint the line. Marry the right person, and you strengthen it. This is language borrowed from wine, from dyes, from metalworking, but it's applied to human beings. Dynastic marriage is where this metaphor turns into machinery. Once you believe that power is carried in blood, family trees stop being just stories about who begat whom. They become tools. A clever ruler can use genealogy the way an engineer uses gears. You claim descent from a legendary ancestor, Romulus, King David, the sun goddess, a heroic founder, and suddenly your reign looks inevitable, even sacred. You arrange a marriage with a rival dynasty, and now two bloodlines are fused. Claims to land, titles, and loyalty move along those veins. By the late Middle Ages and early modern period, European courts are basically running ancestry as political technology. Scribes and heralds assemble complex pedigrees for kings and nobles, snaking back through crusaders, saints, and mythical figures. Legal arguments over who rightfully inherits a throne hinge on which branch of the bloodline is older, purer, more legitimate. You see similar logic in caste systems, in clan structures, in the patrician families of ancient republics. Old blood versus new blood, pure blood versus mixed blood. It's all about who gets access to resources, offices, and status. And because humans are humans, anxiety creeps in. If blood is the carrier of legitimacy, then pollution becomes a constant fear. Who counts as us? Who is other? Which unions are celebrated and which are forbidden? Laws against intermarriage, rules about endogamy, obsessions with keeping the line clean. These are social controls wrapped in biological language long before anyone knows what a chromosome is. Now jump forward to the 19th and early 20th centuries. Science starts to crack open the black box. Microscopists look at blood under lenses. Chemists analyze its components. Then, in the early 1900s, Karl Landsteiner and others identify the ABO blood groups. A few decades later, the Rh factor is discovered. Suddenly, blood isn't just red stuff and metaphor. It comes with letters and plus or minus signs. Here's where things get weird. At first, blood typing is a medical revolution. You can transfuse safely. You can manage pregnancy risks. You can solve paternity disputes with better tools. But those old cultural habits don't vanish just because we learned some new letters. Instead, a lot of people do what humans always do with new information. They retrofit it onto old frameworks. The notion that certain families or peoples have different blood is centuries older than blood science. When ABO and Rh arrive, they slot neatly into that mental architecture. Now, noble blood, or ancient blood, doesn't just feel special in a mystical sense. It can be coded as A, B, O, positive, negative, reducible to a chart. Early 20th century racial theories jump on this. Anthropologists and eugenicists hunt for correlations between blood groups and so-called races or types. They measure skulls, take blood samples, and try to align the results with pre-existing ideas about Nordic, Mediterranean, Slav, Semite, and so on. Most of these efforts are riddled with bias, bad statistics, and circular reasoning. But they leave a legacy, the idea that blood type is a marker of deep, ancient difference between peoples. Rh negative gets folded into this lineage mindset later, once the Rh system is better known. Populations with higher Rh negative frequencies, like the Basque, start to be treated as if they're somehow more ancient, more original, more pure. The script writes itself. These people have a rare blood type. They must be the last survivors of X. Fill in X with Atlanteans, Cro-Magnons, Nephilim, lost tribes, you name it. Underneath that, something subtle is happening. We're blending two very different things, documented genealogy and genetic ancestry. Genealogy, as most people use the word, is about named individuals and paper trails. You trace who your grandparents were and their parents and so on. Maybe you can follow a noble house back 15 or 20 generations. Maybe, if you're a European royal, some parts of your pedigree get stretched into legendary antiquity. But even the most meticulous genealogy is a narrow slice. One thread running through a gigantic fabric. Genetic ancestry is that entire fabric. Every person alive has 2 parents, 4 grandparents, 8 great-grandparents, doubling back every generation. By the time you reach, say, 30 generations ago, around the High Middle Ages, you have over 1 billion slots in your family tree. In practice, the same people occupy many of those slots through cousin marriages and overlapping lines. But the key point is this. Your genome is a chaotic remix of countless ancestors, not a clean transmission of a single bloodline. You can be a documented direct descendant of Charlemagne and share that status with millions of other people. The amount of actual DNA you carry from him might be negligible. Meanwhile, you absolutely carry genetic material from countless unnamed peasants, traders, slaves, migrants, and outsiders who never appear in any royal scroll. When people talk about a royal bloodline, they're really privileging one highly visible branch of a sprawling tangled tree. The biology underneath does not respect those narrative boundaries. Arch negative throws gasoline on that confusion. Because it's simple to state, I'm O negative, she's B negative. It feels like a tag you can hang a story on. And because it's relatively rare in some populations, it invites specialness. If kings and nobles once claimed special status by appealing to mythical ancestors, modern Rh negative narratives do something similar with a lab result. You start seeing claims like, Rh negative people are descendants of an ancient elite, or this blood type proves a separate lineage, or these families protected their negative blood through selective marriage. The logic echoes dynastic politics, a special trait that must be guarded, evidence of distinction hidden in the veins. There's a kernel of historical truth here. Elites really did try to control marriage and inheritance to maintain power and property. Certain lineages really did become overrepresented among rulers. Some isolated communities today really do have unusual frequencies of certain genetic traits because of long-term endogamy. But that doesn't map cleanly onto Rh negative the way the stories suggest. The presence or absence of the Rhd antigen is one tiny feature among thousands in your genome. It doesn't track neatly with royal versus commoner, or with pure versus mixed ancestry. It doesn't care about coats of arms. Yet the temptation to treat it like a modern sigil of hidden nobility or ancient purity is very strong, especially in a world where traditional aristocracies have lost most of their overt power, but the mythology of special blood lingers. So the stage is set. You have centuries of people equating blood with legitimacy and purity. You have dynasties using marriage and genealogy as a form of political engineering. You have modern blood science landing on top of that, offering crisp categories and rare variants. And you have a profound gap between how inheritance actually works biologically and how we like to imagine it works socially. Into that gap flow the next wave of ideas. That negative isn't just medically interesting or geographically distinctive, but spiritually charged. That it marks not just a lineage, but a set of traits, psychic ability, heightened intuition, alien ancestry, whatever the culture of the moment is primed to latch onto. That's where we're headed next. If blood has always been a metaphor for power, What happens when people start saying this particular blood factor comes with hidden powers of its own? Once you have a label that feels special, the next step is almost inevitable. People start loading meaning onto it. Spend any time in Rh-negative forums, Facebook groups, or late-night YouTube rabbit holes, and you'll start to see the same lists circulate. They read like a horoscope for a secret tribe. Rh-negative people, you'll be told, tend to have unusual eye colours, often described as piercing, lower body temperature, sensitivity to heat or sunlight, higher than average EQ, heightened intuition or outright psychic abilities, more frequent paranormal experiences or UFO sightings, a sense of not belonging, of being different from childhood, a rare resistance to certain diseases, or a tendency to never get sick at all. Sometimes it gets very specific. Certain sites insist Rh negatives can't be cloned, or that they're universally resistant to HIV, or that they heal faster than other people. Others go even further and claim anatomical differences, extra vertebrae, different skull shape, warrior genes that make them braver, stronger, or more emotionally intense. Laced through all of this is a much bigger claim, one that jumps from personal quirk to cosmic narrative in a single paragraph. We are not fully human. Here the stories fork into a whole mythos. One branch is ancient and biblical. Rh-negative people are said to descend from the Nephilim of Genesis, the offspring of sons of God and daughters of men, or from fallen angels who mated with humans and passed on their foreign blood. Sometimes this gets synchronized with European folklore about fairy blood, elven ancestors, or second sight running in certain families. Another branch is extraterrestrial. The story goes like this. Rh-negative blood is evidence of genetic engineering by aliens. Maybe they created a worker caste. Maybe they spliced their own DNA into ours as part of some long-term experiment. Maybe they left behind a hybrid population that occasionally wakes up to its true origins. Still another branch is Atlantean or Lemurian. A lost civilization with advanced technology is said to have vanished in a cataclysm, leaving behind survivors whose distinctive blood carried traces of their superiority. According to this script, people with Rh-negative blood are the scattered descendants of that vanished high culture. Their gifts are the last faint echo of a once great power. These are not fringe notions for a handful of people. They have books, conferences, podcasts, entire communities built around them. People swap experiences, prophetic dreams that came true, gut feelings that turned out to be right, encounters with ghosts, aliens, shadow figures. They connect their blood type to migraines, to synesthesia, to being empaths who feel other people's emotions more intensely. Notice what's happening here. A lab result that started as a practical concern for pregnancy and transfusion medicine has been transformed into a badge of identity. Rh-negative becomes not just a medical label, but a personality type, a destiny, even a spiritual path. Why does this happen? Part of it is simple math and psychology. Rh-negative is rare enough in many populations that it feels like an exclusive club, but not so rare that you never meet anyone else with it. That's a sweet spot for building community. If 1 in 3 people had Rh-negative blood, it wouldn't feel special. If 1 in a million did, most of them would never find each other. Add to that the experience many people already have of feeling different, socially, emotionally, psychologically, and the blood type becomes a ready-made explanation. It's not just that you're introverted or anxious or imaginative, it's your blood. You're literally built differently. Once that story takes hold, confirmation starts doing the heavy lifting. Imagine you've been told Rh-negative people are more intuitive. You remember the few times you just knew something, and it turned out to be right. Not getting on a train that later broke down, sensing a friend was upset before they said anything, having a dream about an event that then occurred. Your brain flags those hits as evidence. Meanwhile, all the times your hunches were wrong, the awkward texts you sent because you felt a vibe that wasn't there, the lottery numbers you thought were lucky, the person you misjudged completely, those slide quietly into the background. This is confirmation bias. We notice and remember events that support what we already believe and forget or downplay those that don't. Placebo plays its part too. If you're told your blood type makes you more resistant to illness, that belief can change how you experience your body. You might feel less anxious about every symptom, which can actually reduce stress-related complaints. You might interpret a mild flu as, "I barely got sick," reinforcing the idea of special immunity. Belief doesn't rewrite your genes, but it can absolutely shape perception. There's also a storytelling effect. Online communities are self-selecting. People who feel nothing special about their Rh negative status are less likely to join a group dedicated to it, and even less likely to post about it. The people you hear from are disproportionately those who have unusual experiences, or at least frame their experiences that way. Over time, the shared narrative drifts. The ordinary gets edited out, the extraordinary gets amplified. That doesn't mean everyone is lying. It means that when you gather people who are already primed to see their lives through an Rh negative lens, their anecdotes naturally stack up in a way that makes the pattern feel stronger than it really is. Here's where we have to draw an important line: the difference between testable and unfalsifiable claims. Some of the ideas attached to Rh negative blood are absolutely testable. You can study whether Rh negative individuals differ on average in IQ, in disease susceptibility, in pain tolerance, in certain psychological traits. You can run large-scale surveys, control for background variables, collect data. So far, when researchers look at Rh negative specifically, nothing like a consistent superpower profile emerges. You don't see a clean, reproducible pattern of "these people never get sick," or "these people all score higher on intelligence tests," or "these people unanimously report paranormal experiences." Individual studies occasionally find small correlations, but when you zoom out, they don't add up to a separate human category. Then there are the unfalsifiable claims: alien soul, Atlantean reincarnation, part angel DNA. These are built in such a way that no amount of evidence can really touch them. If you sequence the genome of an Rh negative person and find nothing outside the normal human range, a true believer can always say, "the alien DNA is in a dimension we can't measure," or "they edited their traces so we wouldn't find them." The claim slides away from any possible test and into pure belief. That doesn't make the belief meaningless. It can be existentially important to the people who hold it. It can offer comfort, community, a sense of purpose. But it does mean it belongs in a different category from "this gene variant alters oxygen processing at high altitude," or "this allele increases your risk of hemolytic disease." Understanding that distinction matters, because without it, everything gets flattened. A rigorously tested medical finding and a late-night forum story about seeing lights in the sky get treated as if they're just two equal perspectives. They're not. One is tethered to methods that can be checked and repeated. The other is tethered to narrative, meaning, and personal experience. And yet, once these narratives are out in the wild, they don't stay neatly labelled. They mix with older traditions and newer technologies. The idea that some people carry otherworldly blood has deep roots in esoteric and occult circles long before the Rh factor was discovered. Mystical orders talk about star seeds, indigo children, blue bloods, in more than just the aristocratic sense. When Rh negative enters the conversation, it's a plug-and-play symbol. You can drop it straight into those existing frameworks as the physical proof they were missing. Then modern media steps in and hits the accelerator. A sensational documentary segment about alien bloodlines sparks a wave of blog posts. A channeled message on a New Age forum insists that 15% of humanity are hybrids, conveniently matching global Rh negative frequencies. A TikTok sound about signs you're not from here goes viral, and blood type gets folded into the list. Each retelling trims nuance, adds drama, and tightens the link between Rh-negative and hidden powers. Over time, it stops feeling like hypothesis and starts feeling like folklore, shared, repeated, embellished, rarely questioned inside the group. This is the world we have to navigate next, where centuries-old esoteric idea about sacred blood collide with algorithm-driven platforms that reward the most extraordinary claims, where a genuine medical peculiarity becomes raw material for spiritual branding, identity, and sometimes outright grift. So how did those older occult traditions set the table for all of this? And what happens when the internet takes a slow burn myth and turns it into a feedback loop running at the speed of virality? To really see how Rh negative blood got drafted into this spiritual drama, we have to zoom out from hematology and into a very different kind of history. Long before anyone knew what an antigen was, occult traditions were already obsessed with blood as destiny. Alchemy, Hermeticism, and certain strands of Western esotericism all played with the idea of elect lineages, not just chosen in a metaphorical sense, but carrying a different spiritual voltage in their veins. Medieval grimoires talk about families of the art. Rosicrucian and theosophical writings hint that certain bloodlines are better conduits for higher forces, more receptive to initiation, more capable of holding light. By the late 19th and early 20th centuries, you get full-blown systems built on this. Theosophy speaks of root races, stages of human evolution, some physical, some etheric, with remnants supposedly lingering in certain populations. Early New Thought and Proto-New Age circles rework older notions of blue blood into cosmic terms. Noble blood isn't just socially elevated, it's spiritually distinct. Esoteric Christianity plays with Christed blood, the idea that spiritual transformation literally alters the subtle properties of your physical body. All of this is a kind of pre-internet operating system, waiting for new plugins. When Rh-negative finally shows up on the scene, it arrives as the perfect patch. Here is a measurable difference in blood, rare enough to feel special, invisible enough that you can project anything onto it. It slips neatly into frameworks that already believe some people are wired for magic, contact, or awakening. New Age thought adds another layer. From the 1970s onward, it popularizes concepts like starseeds, souls from other star systems incarnated on Earth, indigo and crystal children, waves of spiritually advanced beings, often depicted as sensitive, psychic, misunderstood. Vibrational frequency, the idea that people, places, and even DNA can vibrate higher or lower, depending on their spiritual development. Once you're thinking in that language, biology becomes a script. Your aura, your chakras, your soul contract, your past lives. They're all said to leave traces in your physical form. Eye color, birthmarks, health issues, unusual reactions to drugs or vaccines. These get recoded as signs of what you really are. Rh-negative slides right into this, not just as a lab result, but as evidence of your placement on some cosmic map. Books and channeled texts start making the connection explicit. An entity speaks through a medium and declares that people with negative blood are keepers of Atlantean codes. An author claims that angelic lineages engineered a protective mutation into human blood. A small spiritualist publisher releases a paperback arguing that H-negative carriers are the physical descendants of the biblical sons of God. None of this is peer-reviewed. It doesn't need to be. Within esoteric and New Age frameworks, truth isn't primarily established by controlled experiment. It's validated through resonance. Does this story feel true? Does it light you up? Do your synchronicities spike when you read it? If the answer is yes, biology is no longer just about red cells and proteins. It's destiny, mission, proof you're not just another mortal stumbling around in the dark. Then the web arrives and hits fast forward. In the early 2000s, Rh-negative chat rooms and bulletin boards begin to appear. A few semi-obscure books and articles are scanned, reposted and quoted out of context. A single speculative line, perhaps Rs-negative blood is a remnant of an older non-terrestrial lineage, gets screenshot, rephrased and repeated until it hardens into fact inside those circles. Forum threads do a lot of heavy lifting here. Someone posts, "I'm Rt-negative, have always seen spirits, hate crowds, and get cold easily. Anyone else?" 10 people reply with, "Same." 3 share long stories about premonitions or near-death experiences. One person remembers something from a conspiracy book and mentions Nephilim. Another drops in a channeled message about hybrid souls. By page 5, a pattern has emerged from anecdotes. By page 10, that pattern is being treated as a profile. Now add early YouTube and blogs. An amateur researcher uploads a video titled Rh-negative blood, proof of alien origin. They throw in some real numbers, global prevalence rates, basic genetics terminology, and then leap straight into ancient aliens, secret rituals, and forbidden knowledge. The comments fill up with people checking in. "O negative here. I always knew I was different." "Be negative and I've been seeing 1111 everywhere." On another corner of the internet, a New Age blogger writes a series on starseeds and includes a bullet. Many starseeds choose Rh-negative blood for its unique energetic properties. There's no citation. It doesn't matter. That line gets copy-pasted into other blogs, folded into listicles, turned into Pinterest graphics, and years later, TikTok slides. Over time, a standardized mythos emerges almost accidentally. Rh-negative people are empaths. They're psychic. They're often night owls. They have trouble with authority. They get static shocks. They dislike strong sunlight. They have green or blue eyes, or eyes that change colour. They're more prone to lower blood pressure, to vivid dreams, to feeling like old souls. Some of those traits conflict. Some are statistically trivial, but memetically that doesn't matter. A meme, in the original sense Richard Dawkins meant it, is simply a unit of cultural transmission. Rh-negative equals starseed is a meme. So is Rh-negative people can't be cloned. So is Rh-negative blood is allergic to the human race. Memes don't spread because they're true. They spread because they're catchy, emotionally charged and easy to remember. Repetition is key. You see the same list of Rh-negative traits on 5 different sites, hear it in 3 different videos, read personal testimonies that seem to confirm it. The human brain is wired to equate familiarity with truth. The more often you encounter a statement, the more likely you are to feel, at a gut level, that it must be real. Anecdotes pour gasoline on that fire. Every time someone comments, "I'm Rh-negative and I fit 8 out of 10 traits," it reinforces the pattern. Nobody's collecting data on the millions of Rh-negative people who don't feel psychic, don't see ghosts, don't have weird health reactions. Silence doesn't go viral, stories do. There's also a powerful belonging effect. For someone who has always felt out of place, stumbling into a corner of the internet that says, "You're different because you're part alien/angel/Atlantean," can be intoxicating. Suddenly your loneliness or sensitivity isn't a bug, it's a feature. You're not broken, you're advanced. You didn't fail to fit in. You were never meant to. This is where identity, spirituality, and biology fuse. Blood type isn't just a medical note in your file, it becomes your origin story, your proof of membership in a scattered tribe of hidden initiates. And once your self-understanding rests partly on that story, you're incentivized to defend it, share it, refine it. To make it feel solid, people reach for scientific language. Rh factor, antigens, recessive genes, hybridization, immune response. Terms pulled from real biology get woven into narratives that have very little to do with how those concepts actually work. You'll see claims like, Rh-negative is a genetic anomaly that can't be explained by evolution. Negative blood types are recessive because they were added later from an outside source. Doctors admit they don't know where Rh-negative came from, so it must be alien. Rh-negative mothers reject Rh-positive fetuses because their bodies know they are not the same species. Each of these lines contains just enough technical vocabulary to sound grounded. They reference real phenomena: rarity, recessive inheritance, hemolytic disease. Then they pivot into speculative leaps: aliens, angels, non-human, DNA. To someone without a background in genetics, the difference between this is biologically odd and this contradicts everything we know about human evolution is easy to blur. And algorithms don't care which is which. They care about watch time, clicks, engagement, content that says, Rh-negative is a mildly interesting population. Distribution quirk with some clinical implications doesn't get shared. Content that says, the medical establishment is hiding what Rh-negative really means, does. This is how we get to the next stage. Suspicion. Once a narrative has crystallized that Rh-negative equals hybrid, chosen, different, the absence of official confirmation starts to look meaningful. If doctors don't talk about energetic properties of blood, it's not because that's not how medicine works. It's because they're supposedly hiding something. If textbooks don't mention Atlantean ancestry, it's not because there's no evidence for it. It's because they removed it from the record. On forums and in videos, a familiar question begins to echo. If this is all nonsense, why won't they address it? Why don't they tell us what's really going on with Rh-negative blood? And that question is dynamite, because it shifts the story from personal identity to systemic cover-up. It's no longer just, I feel special because of my blood. It's, I'm special, and powerful institutions are invested in keeping that truth from me. That move, from myth to mistrust, from spiritual reinterpretation to political conspiracy, is where the arch-negative enigma stops being a quirky New Age sideline and collides full force with a broader culture of skepticism towards science, medicine, and authority. That's where we're headed next. How does a medical footnote become fodder for a conspiracy of silence? Who exactly is supposed to be hiding what, and why? And when we look closely at those claims, what do we actually find? When people start talking about a conspiracy of silence around Rh-negative blood, they're rarely inventing the pattern from scratch. They're plugging Rh-negative into a pre-existing template, a story structure that's been used on everything from Atlantis to vaccines to the moon landing. That story usually has 3 parts. First, there's hidden knowledge, the idea that somewhere, in a locked archive or a classified file, there are documents that explain it all. The origin of Rh-negative, the real reason hemolytic disease happens, the secret experiments done on rare blood types. The information exists, but the public doesn't get to see it. Second, there are powerful gatekeepers, governments, pharmaceutical companies, elite universities, secret societies, ancient bloodline families. Pick your flavor. They're the ones supposedly deciding what you're allowed to know, filtering reality before it reaches you. Third, there are brave outsiders. The forum researcher, the independent YouTuber, the whistleblower doctor, the channeled entity. They're framed as the ones telling you the truth, often at great personal risk, in defiance of the system. Once you've seen that template, you start recognizing it everywhere. The plot can be swapped onto almost any mystery. UFOs, pyramids, lost civilizations, psychic phenomena, Rh negative is just the latest guest star on a very old stage. But the reason that structure works isn't just that people are gullible or bored. It works because there is real historical fuel for mistrust, especially around medicine and human bodies. Think of the Tuskegee syphilis study in the United States, where black men with syphilis were deliberately left untreated for decades so researchers could observe the natural course of the disease. They were lied to, exploited, and harmed in the name of science. Think of forced sterilization programs that targeted disabled people, indigenous communities, and poor women in multiple countries well into the 20th century, often justified by pseudoscientific ideas about eugenics and improvement of the population. Think of radiation experiments on unwitting patients, unethical drug trials in low-income countries, cases where pharmaceutical side effects were minimized or hidden until lawsuits forced disclosure. If your grandparents lived through even one of those scandals, if your community has stories of being used as test subjects or lied to by doctors, trust the science doesn't land as a neutral statement. It sounds like a demand from the same kind of people who caused the harm in the first place. Layer onto that everyday experiences, rushed appointments, confusing jargon, arrogant specialists, medical errors. To many people, the health system doesn't feel like a benevolent, transparent machine. It feels like a black box that occasionally spits out bills, prescriptions, and verdicts about your body. So when someone already suspicious hears that, Rh-negative blood is relatively rare. Doctors pay special attention to it in pregnancy. There's a serious condition where a mother's immune system attacks a fetus, and you need an injection to prevent it. It can easily click into that older narrative. My blood is different, the stakes are high, and the people in charge aren't telling me everything. Then you run into another friction point, the way institutions actually communicate. Science, at its best, lives in uncertainty. Papers are full of hedges. May suggests further research is needed. Medical guidelines change as new evidence arrives. A treatment that was standard in the 1980s might be abandoned now. A risk once thought minor could turn out to be substantial, or the reverse. From the inside, that's a feature. It means the system is self-correcting. From the outside, it can look like shifting stories. Eggs are good for you, then bad, then good again. Masks don't help, then they do. Hormone replacement therapy is a miracle, then it's dangerous. Then it's recommended again for specific groups. Each pivot, however justified by new data, feels like a little betrayal. So were you wrong before, or are you wrong now? Add legal liability to the mix. Hospitals, research institutions, and drug companies exist in a world of lawsuits and regulatory oversight. That environment incentivizes careful, sometimes painfully cautious language. Consent forms are dense with warnings. Public statements lean away from anything that could later be framed as a promise. No guarantees, no absolutes, no grand unified theories. So when someone asks, why does Rh negative exist at all, they're unlikely to get a poetic sweeping answer. They'll get something like, it's a genetic variation in the presence or absence of the D antigen on red blood cells. It likely arose through mutation and persisted because it conferred no major survival disadvantage in most environments. Clinically accurate, emotionally unsatisfying. People looking for meaning hear that and feel an absence, not just of information, but of narrative. No origin myth, no why me, no hint that anyone is even interested in the deeper story of their difference. Into that vacuum step the outsiders, where official channels say, we don't fully know the evolutionary history of this trait. Conspiratorial narratives say, they know exactly where it came from, and that's why they're so quiet. Where scientists say, several hypotheses are being explored, YouTube says, the evidence clearly points to X, and here's why they'll never admit it. the very things that make science epistemically honest, admitting uncertainty, revising positions, avoiding grand claims, become, from the outside, proof that someone is hiding something. No simple answer gets reinterpreted as no answer we're allowed to tell you. You can watch this play out in real time with R negative. Someone Googles around, finds basic medical info on hemolytic disease, a few statistics on global distribution, maybe a dry paragraph on inheritance. Then they go to a forum and find a 2,000-word post connecting Rh negative to Nephilim, royal bloodlines, secret fertility programs, supposed military interest in rare donors. One side feels like a pamphlet, the other feels like a thriller. The human brain has a bias toward agency and intention. If something is unusual and important, like a blood trait that can endanger pregnancies, our pattern-seeking machinery wants there to be a conscious reason behind it. Someone designed it. Someone is using it. Someone benefits if we don't understand it. When that hunger for intention meets bureaucratic capacity, slow-moving research, and cautious communication, conspiracy of silence starts to feel plausible. But this is where we need to pause and do a reality check. If there really were a coordinated effort to hide the truth about Rh negative blood, you'd expect to see certain things: restricted access to basic facts, classified studies, gag orders, suppressed datasets. Instead, what we actually find is an avalanche of openly available information. You can look up the molecular structure of the RHD protein in public databases. You can read original research papers on Rh antigen genetics, fetal maternal incompatibility, and population frequencies by region. Medical textbooks explain exactly how and why rhodiimmune globulin is used, what can go wrong without it, and how protocols have evolved. Population genetics studies discuss proposed evolutionary scenarios for the spread and maintenance of Rh negative alleles. They're not front-page news because they're technical and frankly a bit dry, but they're not hidden. None of this means that every question is answered. It doesn't mean the scientific story is complete or that institutions are always transparent or always benevolent. History shows they're not. What it does mean is that when we talk about a conspiracy of silence around Rh negative, we're mostly talking about something subtler than a top-down cover-up. We're talking about a mismatch between the kind of meaning people want from their biology and the kind of information science is built to provide. Between the messy, evolving, technical reality of research and the clean, dramatic narratives that spread online. In the next section, we'll drill into that mismatch more directly. We'll put specific claims side by side with what's actually in the literature, what science can verify about Rh negative blood, what's still an open question, and where the leap from mystery to mythology really happens. So let's do the side by side the internet almost never shows you. What can science actually say about Rh negative blood with a straight face? And where does the story turn into guesswork, embroidery, or pure fantasy? Start with the solid ground. We know what the Rh system is, biologically. On the surface of your red blood cells, there are structures made of protein and sugar, antigens. The Rh system is one family of those antigens. The most important one for medicine is called D. If your red cells carry the D antigen, you're labelled Rh positive. If they don't, you're Rh negative. We know what gene codes for that protein. It's called RHD. We've sequenced it. We know where it sits on chromosome 1. We can point to specific mutations, deletions, and rearrangements that knock it out and produce Rh negative blood. In some people, the gene is completely gone. In others, it's present but broken in predictable ways. We know how it's inherited. You don't get Rh negative from a past life or a meditation practice. You get it from your parents' DNA. The simplest version is this. Rh negative behaves like a recessive trait. If you have 2 non-functional copies of Rhd, you're Rh negative. If you have at least one functional copy, you're Rh positive, though there are rare, weak D and partial D variants that complicate the picture at a technical level. We also know why doctors care. When an Rh negative person is exposed to Rh positive red blood cells, their immune system can learn to recognize the D antigen as foreign and produce antibodies against it. In transfusion medicine, that can cause dangerous reactions if the mismatch is ignored. In pregnancy, the stakes are higher. If an Rh negative mother carries an Rh positive fetus, fetal red cells can cross into her bloodstream, especially during birth or procedures. Her immune system may respond by making anti-D antibodies. In a first pregnancy, that often doesn't cause obvious harm. But in later pregnancies with another Rh positive fetus, those antibodies can cross back over the placenta and damage the baby's red blood cells. That's hemolytic disease of the fetus and newborn. Untreated, it can be lethal. We have direct evidence for all of this. You can watch the antibodies bind to red cells in a lab dish. You can measure bilirubin levels in affected newborns. You can see in population after population that giving Rh negative mothers Rhody immune globulin at the right times almost completely prevents sensitization and the disease that used to follow. This is the part of the story that lives in hospital protocols and immunology textbooks. It's not glamorous, but it's rock solid. Now, move one step up the ladder to population genetics. What can we say there? We can measure how common Rh negative blood is in different groups today. Large surveys of blood donors and hospital records tell us that in parts of northern Spain and southwestern France, Rh negative frequencies hit 30 to 35%. In much of Europe, they sit around 15%. In many East Asian, African and indigenous American populations, they're far lower, often under 5%. From those frequencies, and from the structure of the Rhd gene in different people, we can infer some things. We can estimate that the mutations causing Rh negative probably arose multiple times in different places, or spread through population mixing after originating in particular lineages. We can build models that ask, if this allele started at a certain frequency tens of thousands of years ago, and people migrated, split and mixed in ways we know from archaeology and other genes, what distribution would we expect to see now? We can test simple scenarios. Is Rh negative so harmful that natural selection should have eliminated it? The answer appears to be no. Does it confer some obvious massive advantage like sickle cell trait does against malaria? If it did, we'd expect clearer signals in the data. Those signals aren't there in any dramatic way. What we can't do is rewind the tape to a specific valley 40,000 years ago and say, here, on this hillside, is where the Rh negative mutation first appeared in one pregnant woman, and she belonged to a secret royal clan. The math doesn't run in reverse with that kind of precision. Population genetics gives probabilities, not movie scripts. It can say things like, given what we see now, these evolutionary paths are plausible, these are unlikely, and these are essentially impossible. It cannot name individual ancestors, attach them to specific ancient tribes with no written records, or prove that a blood factor came from Atlantis, Sirius, or a hidden underground civilization. And that's where a lot of the popular Rh negative narratives leap off the map. You'll often see a chain that goes like this. Rh negative is rare. Some royal families are rumoured to have high rates of Rh negative. Some people on UFO forums say Rh negative folks feel different, or not from here. Therefore, Rh negative is the bloodline of ancient rulers / Nephilim / extraterrestrials, preserved through history. But biologically, a single blood group factor simply doesn't carry that kind of story on its back. Blood group antigens are tiny pieces of an enormous genetic puzzle. Two people can both be O negative and be wildly different in ancestry, appearance, and overall DNA. two others can have almost identical genomes except for the RHD region and end up on opposite sides of the Rh positive/Rh negative line. You can't look at one antigen and declare someone royal any more than you can look at their eye colour and declare them a time traveller. Royal lineages, to the extent they're biologically real at all, are defined by complex patterns of descent across thousands of genes and many generations of social history. Extraterrestrial origin would leave hallmarks across the entire genome, huge swathes of sequence with no match in any known earthly organism, not just a missing antigen that fits neatly into our existing mammalian biology. So when someone claims science has proven that Rh negative blood is non-human, they are at best misunderstanding the evidence. At worst, they're counting on you not checking. Which brings up the obvious question: how do you check? One way to think about information is in layers. At the core, you have primary research, actual studies published in scientific journals. These are where new data enter the world: genetic sequences, clinical trial results, population surveys. They're written in dense technical language. They're not always right. Some get overturned or refined, but they're the closest thing we have to what was actually observed. Around that core, you have synthesis and consensus. Review articles that summarize dozens or hundreds of studies, position papers from professional societies, guidelines that say, given the totality of evidence, here's how we should treat Rh negative pregnancies. These documents are slower to change. They reflect many separate teams trying to replicate and challenge each other's findings over years. Wrapped around that, you have interpretation and narrative, popular science books, documentaries, YouTube channels, forums, blog posts. Some are careful and honest about uncertainty. Others cherry-pick. They cite one small study out of context while ignoring 10 larger ones that say something different. So when you encounter a claim, Rh negative people never get HIV, Rh negative blood came from a lost star race, doctors admit secretly that Rh negative is a different species, ask some basic questions. Is there a primary study behind this, or just anecdotes? If there is a study, how big was it? Was it replicated? Do review articles or textbooks mention this effect, or is it only ever seen on fringe sites? Who benefits if I believe this? Is someone selling a supplement, a seminar, a book, a sense of special destiny? And crucially, has the idea passed through peer review? That process is far from perfect. It doesn't catch every error. It doesn't magically purify motives, but it does mean at least a few qualified people have kicked the tires before something is added to the scientific conversation. In the case of Rh negative blood, the core biology has been through that process thousands of times. The mystical add-ons, hidden powers, psychic gifts, alien ancestry, live almost entirely in the outer layer, where peer review is replaced by upvotes, ad revenue, and book sales. That doesn't mean you have to stop being curious. It doesn't mean every unsolved piece of the art puzzle is boring or trivial. There really are open questions about how and why the Rh system evolved the way it did, and why certain regions show such high frequencies of Rh negative. But when the evidence is thin and the stories are thick, something else is going on. We're not just looking at data, we're looking at a mirror, at how people use biology to talk about identity, power, belonging, and otherness. And that's why, even in the face of clear, accessible science, the myth of the Rh negative enigma doesn't dissolve. It adapts. It incorporates new findings, rephrases old claims, shifts from they're hiding the facts to they're twisting the facts. The question then isn't just what is true about Rh negative blood. It's why does this particular trait attract such heavy symbolic weight, and why do so many people want it to mean more than a protein on a cell? That's where we're headed next. Part of the answer is built right into the numbers. Rh negative is rare. Not vanishingly rare, but rare enough that most people will go their whole lives without thinking about it, and then suddenly encounter it in a blood test or a pregnancy screen and go, wait, what? I'm what? And the human brain does not like stray facts that just sit there. We are pattern-making, meaning-seeking creatures. Give us a trait that's uncommon, a name that sounds vaguely technical and ominous, and a whiff of scientists don't fully know why this distribution looks like it does, and the story engine in our heads lights up. If you're Rh negative, that engine is pointed inward. You already know what it feels like to be outnumbered in a room, whether that's your politics, your religion, your personality, or just the way you look. Being told your blood type is literally negative, and that only a minority of people share it, taps directly into those circuits. Suddenly, a string of childhood experiences, feeling different, feeling like you didn't fit, feeling unusually sensitive or sick or intense, can be reinterpreted through a simple lens. Oh, that's why my blood is different. It's not that the belief causes the experiences. The experiences were there. The label offers a narrative that stitches them together. That's the first layer of the appeal, identity. In a fragmented, hyper-individualized world, identity markers have become a kind of psychological anchor. Astrology signs, MBTI letters, Hogwarts houses, neurodivergence labels, online fandoms. They all serve the same basic function. They tell you who your people are and why you've always felt the way you have. Raw negative, with its mix of medical seriousness and evolutionary mystery, slips neatly into that niche. It's a blood group, but it can be treated like a tribe. Then comes the second layer, the narrative shape. Stories that really grab us tend to have a similar skeleton, a hidden difference, a world that doesn't understand, a secret history, a prophecy, or a buried truth, or a forbidden archive. And at the centre, someone who discovers they are not just random. They are chosen, marked, part of something older and bigger. Harry Potter, the X-Men, Star Wars, every YA fantasy where the weird, lonely kid finds out they're the heir to a forgotten kingdom. If you strip away the special effects, a lot of Rh negative content online is just that story told in biological language. The mystery box is your blood. The forbidden knowledge is the claim that governments, doctors, or shadowy elites are hiding what Rh negative really means. The heroic self-concept is the move from I don't quite fit in to I am a guardian/starseed/remnant of an ancient race. Once that narrative frame snaps into place, almost any new piece of information, an article on genetics, a news story about royal families, a half-remembered anthropology factoid, can be slotted into the plot. Something contradicts the myth? That's not a problem. That's a twist. The cover-up thickens. The villain gets bigger. You can see why this is hard to dislodge with a PubMed link. You're not just arguing about alleles and antigen frequencies. You're tugging at a story people have wrapped around years of their life, sometimes decades. A story that explains their isolation, their health struggles, their vivid dreams, their sense that ordinary life is too small. And human beings will defend a story like that with everything they've got. There's also the social angle. Once you have a trait that feels special and a story that frames it as meaningful, you get communities, forums, Facebook groups, Discord servers, subreddits, YouTube comment sections. People come in with questions. Anyone else O negative and have insomnia? Do any other Rh negatives feel weird around crowds? Is it true we can't be cloned? Other people answer with their own experiences. Some of it's mundane, some of it leans mystical. Over time, the group starts to develop its own mini-culture, its own folklore, its own things we all know, its own enemy figures and trusted sources. Belonging, for many of us, is a stronger drug than correctness. If the cost of belonging is accepting that your blood type ties you to fallen angels or Atlantean priest kings, some people will gladly pay. The payoff, being listened to, being believed, being told that your struggles are part of a larger pattern is immense. And within these groups, the more dramatic versions of the story tend to rise to the top. A post saying, "I'm out negative and honestly it hasn't affected my life at all," is the equivalent of a flat line. A post saying, "I discovered my bloodline goes back to Merovingian kings and I can sense earthquakes," is an explosion of engagement. Which brings us to the algorithm. Most of the platforms we use are not neutral bulletin boards. They are attention funnels optimized for one outcome: keep you looking. Content that is emotionally charged, identity relevant and easy to understand gets more clicks, more comments, more shares. Content that is nuanced, hedged with uncertainty, and full of maybe is comparatively dull. Scientists still debate the precise evolutionary forces that shaped Rh allele frequencies is invisible in that environment. Doctors are lying about alien bloodlines is rocket fuel. The algorithm doesn't know or care what's true. It just knows that one of those titles makes thumbs pause and pupils dilate, so it serves more of that, to more people who have even a glancing interest in Rh negative blood types, conspiracy, or esoteric spirituality. The result is a feedback loop. People who feel different are shown videos that tell them their difference is cosmic. They click. The system learns. It recommends even more extreme versions of the same idea. Communities form around those ideas, feeding the content stream with testimonials and elaborations. Skeptical or moderate voices are harder to find, not because they don't exist, but because they don't perform as well. From the outside, it can look absurd, like a house of cards that should collapse the moment you breathe on it. From the inside, it feels like confirmation after confirmation. Every search leads you to more sources who agree. Every video says, "You're not crazy. They're hiding something." So the enigma persists for three intertwined reasons. Psychologically, because rarity begs for meaning and we are meaning-hungry animals. Narratively, because Rh negative can be turned into a personal epic about hidden lineage and spiritual destiny. Socially and technologically, because our online ecosystems are built to amplify exactly this kind of charged, simple, identity-heavy story. None of this means everyone drawn to these ideas is foolish, or that curiosity about Rh negative is inherently misguided. Very often, underneath the alien lore and royal charts, what you actually see is a search for significance and a demand to be taken seriously by systems that feel cold and distant. The challenge, and the opportunity, is to meet that search without either feeding the fantasy or sneering at it. Because there is a way to hold on to the sense of wonder, the sense that your existence is wildly improbable and deeply connected to ancient history, without needing to believe that a missing antigen makes you a different species. So in the next section, that's where we're going. How to make peace with the real mysteries here, about evolution, ancestry, and identity, without surrendering to stories that only feel true because they flatter us or frighten us. So let's start with permission. You are allowed to be fascinated by this. You're allowed to look at your lab report, see Rh negative, and feel a little shiver of strangeness. You're allowed to wonder why your blood type is rare, why it clusters in some populations more than others, why it almost derailed pregnancy in the first half of the 20th century. Curiosity is not the problem. Curiosity is the engine that got us from "people bleed" to "we can safely transfuse blood between strangers." The question is what you do with that curiosity. One option is to hand it over to the loudest, most dramatic story you can find. Aliens, angels, ancient priest kings. Another option is to hand it over to authority and never think about it again. The doctor said it doesn't matter. End of story. There's a third path. It's slower. It's less cinematic. But it's the one that actually deepens your understanding instead of just feeding your anxiety or your ego. It starts by coming back to the basics and really owning them. Rh factor is a protein, not an aura, not a soul stamp, not a cosmic barcode, a protein on the surface of your red blood cells. If you have the version of the gene that makes that protein, you're Rh positive. If you have the version that doesn't, you're Rh negative. That's it. The reason it matters medically is extremely specific. When an Rh negative immune system sees Rh positive blood, especially in pregnancy or transfusion, it can get confused and attack. That's the hemolytic disease story. That's why obstetricians care, why there are protocols, why there's an entire pharmaceutical product, Rho-D immune globulin, built around this one antigen. The reason it varies in frequency between populations is more diffuse. Founder effects, ancient migrations, genetic drift, possibly some subtle selection pressures we haven't fully nailed down yet. None of that is as emotionally satisfying as secret hybrid bloodline, but it's also not empty. Each quiet word in that explanation, founder effect, drift, selection, is a door into a whole world of real, testable mystery. And you don't have to take anyone's word for it. You can go look, not just on YouTube, not just in comment sections where the most confident person wins, in places where people are accountable for being wrong. If you're Rh negative, you have a built-in excuse to talk to professionals and ask annoying questions. You can ask your primary care doctor or OB/GYN. What exactly does Rh negative mean for me? Can you show me my full blood group results? Why does this matter in pregnancy but not most of the time? You might be surprised how much they're willing to explain once they realize you're genuinely interested and not just bracing for bad news. You can read explainers that are written for humans, not for specialists. University hospital websites, National Health Service pages, simple articles by hematologists who spend all day thinking about blood but also know how to speak in plain language. If a claim you've seen online can't survive a basic check against those sources, if, say, someone insists doctors never talk about Rh negative and your own doctor spends 10 minutes walking you through it, that tells you something. You can also learn how to read your own lab reports. What's your ABO type? What's your Rh? Do you have any antibodies flagged? If the printout looks like nonsense, ask someone to decode it with you, line by line. Once you've seen how ordinary the data looks, it gets harder to project epic myths onto it. None of this kills the sense of wonder. If anything, it can make it sharper. Because here's the deeper layer of the Rh negative enigma, the one that doesn't need any supernatural garnish. Every blood test you've ever had is a tiny snapshot of a story that runs back through millions of years of evolution, multiple near extinctions, countless births and deaths. The alleles you carry, the specific spellings of those genes, made it through ice ages, plagues, famines, migrations. Your Rh status is one little thread in that tapestry. Seeing it clearly doesn't make it less special, it makes it special in a different way. That's where separating personal meaning from factual assertion becomes crucial. You can say, being Rho-negative makes me feel different, and that story of difference helped me make sense of my life. That's personal meaning. It's about how you experience yourself. You cannot, without evidence, jump from that to Rho-negative people are a different species, or Rho-negative people have powers other humans don't, or Rho-negative blood proves alien ancestry. Those are claims about the world. They can be checked, and when they are checked, they fail. The danger isn't in privately feeling special. The danger is when feelings get smuggled across the border and dressed up as facts. That's when people start refusing medical care, or distrusting every professional they meet, or sorting human beings into real and less real based on a protein on a cell. So when you encounter a bold Rh negative claim, you can do a quick internal sort. Is this about how someone feels? Or is this about how the universe allegedly works? If it's the first, you can listen, empathize, compare notes. If it's the second, you can gently ask, How would we know if that's true? What evidence would change your mind? Because underneath all the folklore, the core questions we started with are still sitting there, quietly patient. What is Rh negative really? A variant in a blood group system that evolved in primates long before humans existed. Why does it vary from place to place? Because populations split, moved, shrank, grew, and mixed, carrying their particular genetic dice rolls with them. Why do stories attach to it so easily? Because it's invisible in daily life, potentially serious in a few specific contexts, and wrapped in the kind of techno-medical language that feels both authoritative and opaque. That combination, rare, consequential, poorly understood by laypeople, is rocket fuel for myth-making. You don't have to extinguish the myth impulse, you just don't have to let it drive. You can let art negative be a prompt instead of a prison, a reminder that your body is full of history you'll never fully know. A doorway into learning about genetics, or anthropology, or the brutal ingenuity of the immune system. You can even keep some poetry around it if you like. You can say, "My blood carries traces of ancient bottlenecks and migrations. I am a statistical improbability walking around in the sun." That's not wrong. That's true for everyone, regardless of the plus or minus on their chart. The trick is to keep your stories tethered, to let them stretch, embellish, resonate, but not snap the cord that ties them to reality. So as we move into the final part of this video, that's the question I want to leave humming in the back of your mind. If Rh negative doesn't make you an alien, or an angel, or a lost Atlantean, what does it make you? What changes about your life, your identity, your sense of meaning? If you accept that your strange blood is both utterly ordinary in biological terms and still part of a lineage that barely survived the worst crises this planet has thrown at it. In other words, once you've stripped away the flattering illusions and the frightening conspiracies, what do you think is really going on here? Here's what I think is really going on. When you peel back the drama, you're left with three intertwined stories. The first is medical, very down-to-earth, very specific. There is a protein. Some people have it, some people don't. When that difference shows up in pregnancy or transfusion, it can matter a lot. For most of human history, we had no idea why some newborns turned yellow, seized and died. We had no idea why some women lost baby after baby and blamed themselves, or curses, or fate. The Earth system turned that nightmare into a solvable problem. Once we understood what negative actually meant on a blood cell, we could predict the risk. Once we understood how the immune system was getting confused, we could trick it with a carefully timed shot. The thing that used to look like a curse became a calendar entry. Come in at 28 weeks, and again after birth, get your injection, go home. That part of the story is not mysterious anymore. It's still serious. It can still be life and death if ignored. But it's mapped. It's something lab techs and obstetricians and hematologists deal with every day, often so smoothly that you never even know how close you came to the edge. The second story is population history. Why does negative cluster in the Basque region, in parts of Western Europe, in a few other pockets? Why is it rare in East Asia, rare in much of Africa, rarer still in some indigenous populations? Because populations are not evenly stirred soup. They're little clumps and streaks and swirls left over from glacial refuges, founder events, isolation on mountain ranges and islands and in language groups that stayed put while others moved. When a small group carries a rare variant and then gets lucky, good land, good timing, fewer epidemics, that rare variant can ride the wave of expansion. When a different group gets unlucky, famine, war, disease, that same variant can disappear as if it never existed at all. Rh negative is one of those variants that got lucky in some places and not in others. Mix in the last 10,000 years of farming, herding, city building, empire, slavery, colonization, and you get the patchy modern map we see today. Is there some secret adaptive advantage hiding in there? Something that made our negative slightly better or worse in certain environments? Maybe. There are hypotheses. There are hints. There is not, right now, a smoking gun. So the honest answer, the one that frustrates people looking for a single dramatic explanation, is founder effect, drift, migration, and maybe some subtle selection we haven't pinned down yet. The same forces that shape countless other genes you've never heard of, because nobody made a TikTok about them. That's the second story. Long, slow, impersonal. Powerful, precisely because it doesn't care who you are. The third story is psychological and cultural. You live in a world where you can have your blood typed in an afternoon, but you can't have your life typed. There's no neat plus is minus for where do I belong, or why do I feel different, or why did these bad things happen to me and not to someone else. At the same time, institutions that used to feel solid, medicine, government, religious authority, have cracked for a lot of people. Scandals, contradictions, profit motives, rushed appointments, 5-minute explanations in fluorescent lit rooms. It's not crazy to feel suspicious. It's not irrational to feel like someone, somewhere, is not telling you the whole story. Into that gap between personal uncertainty and institutional mistrust drops this tiny, potent fact. You have a rare blood type that doctors only mention briefly, that can hurt your baby if something goes wrong, that shows up more often in mysterious mountain people and weird internet subcultures. Of course, stories glue themselves to that. Humans hate meaningless difference. If we feel different, we want that difference to mean something, preferably something flattering. I am defective is one way to make sense of pain. I am chosen is another. So you get two main flavors of myth. I am special because my blood is from somewhere else. Aliens, Nephilim, Atlantis, lost high priests, anything that lifts me out of the ordinary human mess. And I am targeted because my blood is from somewhere else. Governments hiding my abilities, medical systems secretly harvesting my genes, elites trying to suppress my kind. If you've had weird medical experiences or felt alienated or been burned by authority, those myths can feel like finally found context, like someone has pulled back a curtain. The problem is that when you check them against the first two stories, the medical reality and the population history, they crumble, not because science is boring and unimaginative, but because it's already done the hard work of asking, if this was true, what patterns would we see? What experiments would confirm it? If Rh negative were alien, we'd see it in the genome as something wildly out of place. We don't. If it conferred consistent psychic abilities, you'd see it in big controlled studies, because someone would have cashed in on that by now. You don't. If medicine was secretly obsessed with you, you'd see research funding, clinical protocols, entire specialties clustered around your blood. You don't. You see exactly the level of attention you'd expect for a medically important but pretty mundane antigen. So from my perspective, what's going on here looks like this. A real, non-trivial medical issue that used to kill babies and doesn't have to anymore. A real, fascinating, evolutionary and demographic story that shaped where you and your ancestors ended up, and layered on top of that, a very human fog of projection, fear, identity, mistrust and creativity, spinning those facts into much bigger, wilder narratives than the evidence can support. That doesn't make you foolish for being drawn to those narratives. It makes you human. You're allowed to like the poetry and still insist on the data. You can say, it feels meaningful that my blood is rare, and also, I accept that rare does not equal non-human. You can be skeptical of institutions and still, on balance, trust that your OBGYN is not part of a centuries-long plot to suppress your Atlantean powers. You can hold on to the wonder, the sense that your body carries traces of deep time, bottlenecks, migrations, without needing to leap to, therefore I am not like other humans. If there's a conspiracy here at all, it's the conspiracy of our own brains to avoid the ordinary truth, that every one of us, positive or negative, common or rare, is already the product of an absurdly unlikely chain of survival. That we're all walking monuments to ancestors who made it through events far more brutal than anything in a YouTube title. That should be enough. It usually isn't. But it could be. Now, that's my read. That's what I think is going on when you zoom out and connect the dots between immunology, population genetics, folklore, and the modern attention economy. But you're the one sitting there with your own lab results, your own family stories, your own late-night rabbit holes. So I want to hear from you. If you're Rh negative, how did you first learn? Did it change the way you saw yourself? Did anyone in your life attach a story to it? Mystical, medical, or both? If you're Rh positive, have you run into these myths before? Do you see similar patterns around other traits, neurodivergence, rare conditions, ancestry tests? Drop your perspective in the comments and try to keep it respectful. You don't have to agree with me or with each other, but this topic goes off the rails fast when it turns into, I'm more evolved than you, or you're all idiots for believing X. Let's not do that. And since I'm genuinely curious about who's listening, tell me where you're watching from and what time it is there right now. I love seeing the spread. 2 a.m. insomniacs in London, lunch breaks in Sao Paulo, bus commutes in Manila. Put your pin in the map. If this video helped you untangle some of the noise around Rh negative, or at least gave you a new way to think about it, take a second to hit like. It tells the algorithm this was worth someone's time. If you have a friend who's deep into Rh negative lore, or who just found out their blood type and is spiraling, send this their way. And if you want more deep dives like this, where we take something that looks like a simple fact or a wild conspiracy and trace it back through real history and real science, go ahead and subscribe and turn on notifications so you actually see the next one. I've told you what I think is going on here. Now it's your turn.